start in a Twitter. If you are on Twitter, you can see the Twitter handles for PRiME and ETH are here. And we're also asking you to use the hashtag PRiME Zurich for our joint, for the joint symposium. So just to go through a few things before we start to make sure that everyone has a good experience today. In terms of accessibility, today's symposium is being recorded. Live captioning is enabled. You can display subtitles by clicking on live transcript function at the bottom of the Zoom screen, bottom right of the Zoom screen. The chat function has been disabled. So please use the Q&A function to leave questions for each of the panelists. And we do hope that you leave questions and comments because that'll make the symposium more engaging, more interactive. And if you have any accessibility needs or concerns, please email Angela at ac.zoo@mail.utoronto.ca. And just before we get started, I'd like to thank Christine Mesquita and Angela Zhu for the tremendous work that they've put into organizing this symposium today. And also thank you to Jean-Christophe Leroux for serving as our contact at ETH. So thank you for that. So we're going to begin today with a land acknowledgement. And this is an important week for us in Canada. September 30th is now recognized here as Truth and Reconciliation Day. We wish to acknowledge the land on which the University of Toronto operates. For thousands of years, it has been the traditional land of the Huron-Wendat, the Seneca, and most recently, the Mississaugas of the Credit River. Today, this meeting place is still the home to many Indigenous people from across Turtle Island, and we are grateful to have the opportunity to work on this land. So we have two exciting themes for you today and a phenomenal series of speakers in each theme. We'll begin with a focus on disease in a dish or on a chip models, and we'll then take a short break and resume with our second theme focused on engineering molecular structure for imaging. So on the next slide, we just have a brief overview of today's agenda. So we'll start at 9:15. We've got three fantastic speakers. We won't have any time for Q&A after each talk. We're going to save all of the Q&A and discussion for a session right at the end of the three talks. So we'll have the three talks, and then we're going to have some time between 10:15 and 10:35 for discussion and Q&A. We'll then take a short break, and then we'll be back for our second theme. And again, following the same structure, three talks, and then we'll have time for discussion and Q&A. So I'm delighted to be here today. My name is Christine Allen. I'm a professor in pharmacy with research focused in the area of pharmaceutics and drug delivery. I'm also the Associate Vice President and Vice Provost Strategic Initiatives at the University of Toronto. And in my role as Associate Vice President and Vice Provost Strategic Initiatives, I support the launch and development of large-scale interdisciplinary research initiatives that span the institution. PRiME is one of our leading strategic initiatives. It was founded by Professor Shana Kelly and Stéphane Anger in 2019. Professor Kelly has now unfortunately departed the University of Toronto for Northwestern University, and I have the privilege of stepping in for the next few months during this transition period. Over the past 2 years, PRiME has grown tremendously, and it now represents an important research network that drives collaboration in the area of precision medicine across our institution. So a little bit, I'm going to give you a little bit about who we are, and then our goals within PRiME and the activities that we're engaged in. And then I'll cover a little bit on the Connaught Global Challenge program that we're fortunate to have funding from, which has led to bringing us all together today. So first of all, a little bit about the University of Toronto. We are a large university with a total enrollment of more than 95,000 students. Our student population includes domestic and international students, with international students from 164 countries and regions. We have more than 15,000 faculties. Typically in any one year, our institution is awarded close to $1.5 billion in research funds. In an area like precision medicine, what makes us very special in terms of a place to be or location are our connections to our strong hospital network, as well as our connections to a strong and vibrant entrepreneurship, and commercialization community. We are essentially across the street from some of the top hospitals in the world, such as the Princess Margaret Cancer Center, the Hospital for Sick Children or Sick Kids, and many more. And this gives us access to clinicians who help us identify unmet medical needs, can work with us then to guide the research to ensure it meets those needs, and can lead or champion the translation of our technologies and our therapeutics. We also have a very strong entrepreneurship community with more than 10 campus-based accelerators. And across the street from us is something called the Mars Discovery District, which supports new startups and their scale up. So it's really, from my perspective, the place to be if you're conducting this type of research. In a short time, PRiME has quickly become an exceptional community. PRiME unites biomedical researchers across the University of Toronto to foster collaboration and high impact research. It now includes 85 faculty members and more than 200 trainees across 15 University of Toronto departments. PRiME faculty are world-renowned leaders with a track record of collaborative and translation-oriented research. So to give you a sense of the types of things that PRiME is focused on, our goals: elevating research in drug discovery, diagnostics, and disease biology. We'll hear about some of that research today. Empowering and fostering collaborations across our ecosystem, but also with external stakeholders, including, as I said before, the hospital network, as well as industry. Training multidisciplinary and translational leaders. There's a huge emphasis on training within PRiME. Creating community. I can tell you many of my trainees are part of the PRiME community and have really appreciated, in particular during COVID-19, when things have been shut down at times and it's been somewhat isolating for students. PRiME has continued all of their offerings, which has been fantastic for our trainee community. And of course, PRiME is very much focused on building a biotech ecosystem. PRiME's research strengths include biologics and antibody engineering, high throughput omics, nanomedicine, AI and drug discovery, precision diagnostics, targeted small molecules, biology on a chip, we're covering that today, and high throughput screening platforms, as well as the development of novel therapeutic targets or identification of novel therapeutic targets. So importantly, the symposium today is part of PRiME's successful application to the Connaught Global Challenge Fund. The Connaught Fund supports a number of internal grant programs, including the Global Challenge Program. The Connaught Fund was founded in 1972 when UofT sold the Connaught Medical Research Laboratories for $29 million. The lab had been established in 1914 to produce diphtheria antitoxin. After the 1921 discovery of insulin by UofT researchers, Banting and Best, the lab expanded and began producing insulin as well as other vaccines and antitoxins. 2021 is a special year for us because this is now the 100-year anniversary of the discovery of insulin. So you'll see UofT today, or UofT this year, frankly, sharing a lot on what UofT has accomplished since the discovery of insulin and how that has really helped us to grow our ecosystem. The Connaught Challenge program connects PRiME with a number of different institutions, importantly UCSF, Hebrew University, ETH, National University of Singapore. And so this will include what these funds have allowed us to do is to have a speaker series, and importantly also a PhD and postdoctoral fellow exchange program. We also have the PRiME Scholars Program for undergraduate students. And today, of course, we're kicking off with this joint symposium. So I'm delighted to be hosting today with Professor Jean-Christophe Leroux, who needs obviously no introduction. I heard everyone saying hello today to him when they were joining the call today. He's a dear friend of mine and someone who has very strong ties to Canada. Jean-Christophe is a full professor in drug formulation and delivery at ETH Zurich. He received his Bachelor's of Pharmacy from the University of Montreal, followed by a PhD in Pharmaceutical Sciences from the University of Geneva. From 1996 to 97, he completed postdoctoral training at UCSF and then joined the University of Montreal as an assistant professor. He was promoted to the ranks of associate professor in 2002, full professor in 2007, and he was also the Canada Research Chair in drug delivery. Professor Leroux joined the Institute of Pharmaceutical Sciences at ETH at the end of 2008. And this was, of course, a huge loss for us in Canada, but we were excited to see him take on this new and exciting role. I have to tell you that in the last few years, and even this year again, I'm seeing many new professors that have taken up positions in Canada that have trained with Jean-Christophe. So in some ways, he's still very much here with us. So welcome, Jean-Christophe, and I invite you to introduce our first speaker.
Thank you very much, Christine. So I'm really happy to be here today, although it's only virtual, but we have a great speaker list. So we're going to start with Professor Cornelia Halin, who is a great scientist and great colleague. She has been Associate Professor of Pharmaceutical Immunology at ETH since October 2014. And her research mainly focuses on how leukocytes migrate across the lymphatic endothelium. And an important goal of her research is to identify and validate new adhesion molecules, which eventually could serve as therapeutic targets to modulate immune responses. So the title of her talk today is Blocking Leukocytes Migration for the Treatment of Corneal Transplant Rejection. Cornelia, the virtual floor is yours, and I'm really looking forward to hear your talk. I know you're great speakers, so we're all excited about this. Thank you very much, Cornelia.
Okay, well, thank you very much to Christine and to Jean-Christophe for inviting me to this great event. It's a great pleasure and honor for me to participate and to give the first presentation. As you see from the title, I'm a bit the wild card in this session, I think. So it's not about disease in a dish or in a chip. I will be presenting a lot of in vitro essays or in vivo essays, but I think it has quite a translational component, and I'll be very grateful for your feedback and hope it's insightful. So Jean-Christophe already told you what I'm interested in. So my lab is very much interested in how leukocytes migrate through afferent lymphatics. And you might know that these vessels are present in virtually all tissues of our body, and they exert important functions for tissue fluid drainage, but they're also important for the immune system since they transport antigens and leukocytes from peripheral tissues to one of our perhaps 500 lymph nodes in our bodies, where then immune responses are being initiated. So one cell type that we're particularly interested in my lab are dendritic cells, which also migrate via this route. And you might know that these cells are very important for inducing adaptive immune responses in lymph nodes. And they're present in many of our tissues, in virtually all tissues, but very much in our tissues at the surface of our body, for example, in the epidermis. And for example, if you have an infection, then these cells will get activated. They can take up antigen derived from the pathogen. They will transport it then via lymphatic vessels to the lymph node and then present it on their MHC molecules to a T cells in order to activate cognate T cells for that pathogen of interest. So they kick off the adaptive immune response. That's their physiologic function. But of course, they are also of interest from a therapeutic perspective. I think now in the times of COVID, I don't have to talk much about vaccination, where obviously this process is also very relevant since vaccine can be transported the same route and presented to T cells. And another condition where this process is of importance is actually transplant rejection. So when a foreign tissue is placed into our body, and of course you want to prevent rejection by giving strong immunosuppressives, but in principle this is one of the priming events that would take place in the case of a rejection. Now, in my topic, I want to introduce you to a particular type of tissue transplantation. This is actually corneal transplantation. The cornea, as you all know, that's the upper front part of your eye, and it's a translucent tissue, and it has two main functions. It's important for the protection of the eye and also for the refraction of light. It's also very interesting from a transplantation point of view, since it's actually the most frequently transplanted tissue worldwide. There are many reasons why you might need a corneal transplantation, and they're all linked to blindness, since corneal disease is the fourth leading cause of blindness. And so reasons for doing so might be when this translucent tissue actually gets opaque. for example, through growth of blood vessels and also leukocyte recruitment, and then leading to the breakdown of this translucency. So example, in the context of an infection, if you have injuries or chemical burns, if you have a misshaping of the cornea, or also in the context of repeat transplantations, which occasionally are necessary. But generally, one can actually say that the cornea is a very well tolerated tissue in terms of transplant rejection. Many patients fall into this category. They are low risk patients. That means that they accept these transplants very well. And this is really linked to the fact that the cornea is an immune privileged site. This means that there are a bit different immunologic conditions that are occurring here. And this is very much linked to the fact that the cornea is an avascular tissue. So you typically have no blood vessels and also no lymphatic vessels. So there's not a real communication with leukocytes in blood or with draining lymph nodes, making it a site where rejection is not so frequently induced. So typically, the standard of care after a transplantation would be corticosteroids, which are administered topically for 6 months and then are tapered off. And even so, the rejection rate is really low compared to many other solid organ transplantations. However, there are conditions when patients fall into this high-risk group where the situation is a bit different. And for example, the main factors making you a high-risk patient would be whether if you have already previously rejected a graft, then you will actually have an inflammatory condition present in this cornea. And this may lead, together with other inflammatory conditions or also infections, can lead to the ingrowth of blood vessels into the cornea, or also the ingrowth of lymphatics, which you don't see, but which typically is concomitant with the ingrowth of blood vessels. So under these conditions, this immune privilege is actually broken down because now you have communication with the blood and draining lymph nodes. And actually, in this case, the rejection rate is fairly high. And also the treatment is not, I mean, as you imagine, is not as effective. So typically it again consists of continued topical treatment and often also combined with some systemic immunosuppression. But still, it doesn't really help to reduce this rejection rate. And I think you see it very well here. How vascularization in the bed in which you engraft the cornea affects the transplantation rate. So if in all 4 quadrants, if you have vessels, then you see how this 5-year survival really goes dramatically down. And the same is true with repeat grafting, where of course you have now already an immune response at place. So if one wanted to sum up now this biology of corneal rejection, then I think it's nicely explained here. So imagine you transplant the cornea into an already vascularized and highly inflamed bed. Then in this case, it will be easy to have more leukocyte inflammation and a kind of a movement of the inflammatory conditions into the graft. And this can then lead to the activation of antigen-presenting cells in the graft and also the formation of new lymphatics into the grafted tissue, which leads to migration of dendritic cells via lymphatics to the eye-draining lymph nodes, where then an allo response, so a response against this grafted tissue, is initiated. You will then have formation of activated T cells, which migrate through the blood vessels that are also formed here into the graft and will then attack and induce the rejection process. So if you now had something to block all these different steps, so the lymph angiogenesis, the dendritic cell migration, as well as the activation of T cells, this would obviously be quite an interesting target to work with. And actually, in the course of our experiments working in the fields of lymph angiogenesis and this migration, we came across a molecule which we think is quite interesting in this regard. And this is the activated leukocyte cell adhesion molecule. So we found in an old study, working still with these microarrays, that human lymphatic endothelial cells express this adhesion molecule. It's an adhesion molecule that belongs to the immunoglobulin superfamily, and it has a fairly broad expression pattern. It was previously found on blood endothelial cells. We also here detected it on lymphatic endothelial cells. And it also is expressed on certain leukocytes, including dendritic cells. Furthermore, it has been described on tumor cells and also on certain neurons. Now, the role of ALCAM in all these cells is basically to stabilize cell-cell interactions. And it does it by allowing formation of homophilic interactions, alcam-alcam, as well as heterophilic interactions with other binding partners. So it can stabilize endothelial cell junctions, support leukocyte transmigration, and has also previously been shown to be important for dendritic cells activating T cells. So we started this project working with human endothelial cells, lymphatic endothelial cells, where at the time we could show that if we block ALCAM with commercial antibodies or FC fusion proteins, we can block processes like tube formation or also migration of lymphatic endothelial cells and their ability to close, for example, a cell-free area in a scratch wound assay. We also previously showed in ALCAM deficient mice that they have a defect in a formation of a mature lymphatic network. This is illustrated here by analyzing the diaphragm, which has a lot of lymphatics in the central tendon area in newborn mice. And you see that they have thinner vessels and more branching points in the case of ALCAM, because it seems to prevent or doesn't allow to stabilize larger vessels and to prune this initial network. Also, we found that dendritic cell migration, in this case, looking at the migration of lung dendritic cells to lung draining lymph nodes in a bead assay where we instilled FITC positive beads into the lung and then looked how they arrived in the lymph node. And you see that this was reduced in ALCAM deficient mice. So this made us very interested in this process, but at the time there was no mouse blocking antibody available. And we actually found a way around it without making an antibody, but we found an antibody that was previously published to block a human ALCAM. And we tested it and actually found that it also binds and blocks murine ALCAM, which has about 85% sequence homology. We fused it with an FC fragment to increase its half-life in a therapeutic application. And as you see here, we also looked at its specificity. So looking at endothelial cells in mouse, in deficient or wild-type mice, and you see this antibody nicely binds in the wild-type, but not in the ALCAM deficient mice. So using this antibody, we then tested it in different in vitro assays on mouse ALCAM and could reconfirm that blocking ALCAM also had an impact on scratch wound closure in murine lymphatic endothelial cells. And interestingly, we also now here saw it expressed on dendritic cells and could see that if we block it, it blocks the transmigration of dendritic cells across lymphatic endothelial cell monolayers. And as previously reported, we could also, to a certain degree, reduce T cell proliferation when we, in an in vitro experimental model, looking at T cell induced proliferation when they see antigen on dendritic cells. So it seemed quite interesting that it blocks all these relevant processes. And therefore, we teamed up with the groups of Professor Klaus and Felix Bock at the University Hospital in Cologne. They're actually one of the big reference centers in Germany where corneal transplantations are performed, and they have a very nice animal model established for this. So in this animal model, they try to mimic the high-risk situation in patients by actually first placing sutures into the cornea, which is irritating the cornea of the animals and then induces neovascularization, so formation of blood vessels and lymphatics. And if you now do an allograft, so you graft a cornea derived from Black 6 mice into Bulb C mice, then this will get rejected faster than if it was than if you had not done this with the prior vascularization. So we used this model. And so in this model, this high-risk situation was induced, and then transplantation took place, and treatment with either this ALCAM blocking antibody or with the control FC fusion protein consisted of systemic or topical treatment was given for 2 weeks. So to a bit mimic the patient situation where you just treat for a short time and then leave it for itself. And you see that ALCAM blockade had a significant impact in preventing the rejection and enhancing the survival of these grafts. So that was very interesting for us. And we also looked greater at the details of analyzing these corneas and draining lymph nodes. And we could see that there were actually DCs accumulating in the corneas. However, we did not see an effect on lymph angiogenesis in this model. So we assume that the mechanism of action here is that dendritic cell migration is hampered from the transplanted cornea via the newly formed lymphatics to the draining lymph nodes, and that this accounts for the survival benefit. Now, in collaboration with Klaus and Felix Bock, we also could stain some rejected human corneas, which confirmed that ALCAM is actually expressed also in the vasculature in human cornea. So it's expressed in blood vessels as well as on the endothelium of lymphatic vessels, as shown here by these staining of adjacent sections. And also the MHC positive cells express ALCAM. So we think that this is quite an interesting target for translation. And we have now initiated a new project for which we got funding from InnoSwiss in Switzerland, where we now want to generate better antibodies and also with IP protection, since, as I mentioned, this antibody moiety was already published. Now, unfortunately, I cannot give many details because we haven't filed the patent yet of this study, but I want to finish off by giving you some considerations of what we think has to be considered for making good antibodies for topical applications in corneal transplantation, because actually topical treatment, this is really what is needed according to our collaborators. One issue is, of course, the question of penetration. The cornea has a very tight epithelium, a multi-layered epithelium, so it's quite a tight barrier, and the vessels will be formed here in this stromal part of the cornea. Now, if you want to look at how molecules penetrate this cornea, there are different models at hand. There are corneal tissue culture models. This is something we're testing out now, where you get these corneal epithelial layers formed already on a transmembrane plate, and you can look at permeability. Other studies have worked with intact excised animal corneas and looking at permeability or looking at this in vivo. But obviously, one issue that's already quite clear from the start is that size likely matters. There are many publications on this saying that fragments of antibodies cross the cornea better as compared to full-length antibodies. On the other hand, if one looks at what has already been approved or tested in topical applications in human corneas, there's actually just one therapeutic protein that has been approved. This is recombinant human nerve growth factor that is used for the treatment of neurotrophic keratitis. It's a rather small protein of 13 kilodalton. And there's also, that's perhaps the most advanced other molecule, that's an anti-TNF antibody single chain, which is used for inflammatory diseases of the frontal part of the eye. And this is now in phase 2A studies. There were positive results reported. On the other hand, although these are fragments, there are some curious observations using topical application of entire antibodies, bevacizumab, an anti-VEGF-A antibody that has already been shown by several studies, not just the two ones referenced here, to reduce corneal neovascularizations in inflamed human corneas. So this has, for example, been tested as a treatment to reduce neovascularization prior to transplantation, and effects were seen here, so indicating that also penetration across of an entire antibody must be possible to a certain degree. And in fact, there is now a clinical trial ongoing with topical bevacizumab also in the context of high-risk corneal transplant survival. Even so, I mean, we think that probably the solution will be a fragment. This is again the size of the molecule we have been working with so far, and you see there are many options for how to make these antibodies smaller, retaining their bivalency or making monovalent antibodies. And of course, another issue will be to generate just a new antibody or one of higher affinity, because of course also affinity will matter. Here we're in the lucky situation that we have access to a phage display library from the lab of a former colleague of us, Professor Dario Neri, and we're also doing affinity maturations, obviously, to increase the affinity of our antibodies. So with this, I'm coming to my conclusions. I hope I could show you that ALCAM is an interesting target in the context of corneal transplant rejection because it blocks or has an impact on 3 processes that are presumed to be important for the transplant rejection. And indeed, we could verify this in the mouse model, and think it's also interesting from a translational point of view because it's expressed in the human corneas. So with this, let me come to the most important slide, the acknowledgements. Most of the data I presented were from a former PhD student, Halin Wilroth, and we're currently working busily on the continuation of the project. This is Morgan Hunter and Aline Bauer, and we were helped by many people from the lab and great collaborators, and particularly Felix Bock, Klaus and their teams who are doing all the animal studies for us. And with this, I'm at the end of my presentation, and I'll be looking forward to your questions at the end of the session. So, thank you,
Cornelia, for being right on time. Yeah, I guess, Christine, now maybe you can introduce the next speaker.
Wonderful.
Thank you very much, Cornelia.
Thank you, Cornelia. Fantastic talk. And just again, asking everyone to hold their questions until the end of all three talks. And our next speaker is Professor Milica Radisic from the University of Toronto. She's a professor in the Faculty of Applied Science and Engineering here. Her research program focuses on pursuing molecular mechanisms governing the formation of contractile cardiac tissue in vitro, as well as on practical strategies for treatment of myocardial infarction and heart failure through development of new biomaterials. And today, she'll be telling us about organ-on-a-chip models of COVID-19. Welcome, Milica.
Thank you very much, Christine. It's really a pleasure to be here. I hope everyone can see my screen. All right, and I'll just go like this.
Perfect.
Yeah, so I'll introduce a little bit our BioEyer model that we started from. And it's basically a muscle of a cardiac muscle that looks like a trabecula. That's about 5 millimeters long, about 600 microns in diameter, and we make it from iPS-derived cardiomyocytes. At the end of differentiation protocols, these cells are usually mature, and that was quite obvious some 10 years ago when these protocols were being developed. And using this engineering approach, we were able to contribute to cell maturation quite significantly. So first of all, we microfabricated tissue wells from PDMS into which we could place one suture, and then we would take cardiomyocytes at the end of IPS or human embryonic stem cell differentiation protocols. We would reconstitute these cells in a hydrogel, such as collagen hydrogel, and seed them in the microwells. The cells then start pooling on the matrix. It's a well-known process of gel compaction. They elongate when they come close to the suture, and basically they create something that looks like a trabecula. And this structure was definitely motivated by the native cardiac muscle, where you can see that all cells align with one capillary. What we did then is also we applied electrical field stimulation of ramping frequency increase. And this is really made to mimic things that are happening during development, where the first beats are very slow and irregular, and then they really speed up. And so we found that pushing the cardiac cells really to the limit of their ability, going up to 6 Hertz, led to significant maturation in terms of shape. We get a lot more rod-shaped cardiomyocytes. We get mature calcium handling. We can see that when we block L-type calcium channels with rapamycin, we add caffeine, we see this release, which suggests that the release of calcium is happening from the sarcoplasmic reticulum. Several years later, we replaced this suture by a pair of contracting polymer wires that you can see in this movie. We published that in Cell. The original method we published in Nature Methods. And now we have the tissue hanging in on 2 wires, and we are anchoring them at 2 ends, which leads to anisotropic cell orientation through orientation of these attractional forces. And if we take the stimulation protocol out to 6 weeks, we get another hallmark of cardiac maturation, which is the positive force frequency relationship that you can see here, completely quiescent myocardium when we turn off electrical stimulation, and post-stress potentiation. So all of these are hallmarks of a very mature myocardium. This system is excellent actually for many different models of disease. One of them is cardiac fibrosis. You can see that basically we can use this platform to mimic human cardiac fibrotic tissue, and we get hallmarks such as increase in stiffness of the tissue, and also by proteomics, as we published in Nature Biomedical Engineering. These tissues are very similar in their proteome to basically human myectomies, which are pieces of cardiac muscle that are taken at surgery from patients who have left ventricular hypertrophy and fibrosis. A lot more similar than what the mouse are to the human hearts. In addition, we use the same system to design a model of left ventricular hypertrophy that results from hypertension. And we used cells from 3 affected and 3 unaffected patients. So these are patients, unaffected are those who have high blood pressure, but they don't have left ventricular hypertrophy. And then affected ones are those who have high blood pressure, they end up having hypertrophy, and in some cases even heart failure. You can see that upon 8-month cultivation in this platform, we are able to achieve quite significant genetic hallmarks of left ventricular hypertrophy. And in affected patients, we also lose contractile force, which is a hallmark of this end stage left ventricular hypertrophy. The cells are still alive, they're still there, they're just not beating. And the non-affected patient cells, the tissues made from them are still beating. More recently, we were also able to create a model of drug-induced left ventricular hypertrophy and cardiomyopathy by the use of angiotensin II. And in an unpublished work, we are taking cells from patients who have dilated cardiomyopathy caused by a mutation in sodium channel. These are patients who are followed at Toronto General Hospital, and we can reprogram their blood cells into iPSCs and create these models of cardiac muscle. You can see that the mutated cells derived from the patients, they basically dilated the tissues. They also express a lot less cardiac troponin T compared to corrected controls. This is a monogenic mutation, so they're corrected by CRISPR-Cas9, and they also exhibit much significantly lower contractile force, as well as signs of hyperexcitability by this, what you can see here from action potential recordings. So we have a pretty robust system here for disease modeling. We also wanted to move from basically handmade artisan devices, where these chips are made by hand, to mass-produced devices. And this is really something that's enabled by our new craft facility and collaboration with NRC. Chinghua was one of the co-supervised postdocs with NRC personnel, and we can hot emboss an entire 96 or 24 well plate and move from an A12 chip that can make 8 tissues to something that can make dozens of tissues at a time. In a single step, we place electrodes into the this plastic bottom that's custom made, and then we 3D print the wires from thermoplastic elastomer on top. And this, we had to make some modifications to the wire itself. We moved, as I said, to thermoplastic elastomer with dissolved quantum dots. And so this material exhibits a very stable fluorescence, just like our polymer wires. And it's completely non-toxic to the cells. And it also exhibits very reproducible behavior when it's being pulled on. And so what you can see here are examples of calibration curves that we can use to figure out the contraction force. And they remain also pretty stable, even if you immerse material in culture media for a period of time. And so just to illustrate what this automation does for us, you can see this manual process before to make this sort of A12 chip fitted in a petri dish. It takes about 65 minutes per well, or almost 4,000 minutes per one device to create the platform. And if we automate using these hot embossing methods that we can really perfect through craft, we increase the platform production efficiency by 69,000%, which is a very significant increase. And now we have this better platform. It's a plate. It doesn't have any sharp parts, and basically it doesn't need any pumps or anything like that to run, which means we can take it very easily to CL3 facility. And this is really another strength of University of Toronto and generous support during COVID of CL3 facility. And so now this gives us an opportunity to study how SARS-CoV affects cardiac muscle. And anecdotally, it's quite well known now, and not just anecdotally, but also through clinical trials, that SARS-CoV has tropism for the heart, that it can affect the myocardium, and that there are patients who suffer from cardiac side effects after they have survived COVID and recovered. And so it's really important to understand what's the contribution of direct infection on the myocardium versus contribution of the cytokine storm of the inflammatory response. And so first of all, in our hands, and other people also published on this as well, SARS-CoV-2 can affect cardiomyocytes persistently. This is very different than other coronaviruses that do not have any tropism for the heart. And you can see an experiment here where we take SARS-CoV, we apply them to human iPS-derived cardiomyocytes, and then we can look for viral protein production by Western blotting, or take this supernatant from infected cardiomyocytes and apply them to virus cells to calculate TCID50. And basically, that's how we can tell what's the killing ability of this virus on the new cells. And what you can see here is that, especially the longer the incubation time of the virus with cardiomyocytes, the more we are killing virus cells. And so this is a pretty conclusive proof of infection. And then we took this virus to a BioEyer model. And basically, the overview of the workflow is shown here. We create a tissue for 1 week, and then we didn't take the tissue up to 6-week maturation because we wanted to be able to do these experiments quickly in the interest of time. And then we apply in some groups extracellular vesicles as potential therapeutics that could reverse some of the negative effects of SARS-CoV infection. And we incubated tissues for 1 week, and 2 weeks later, we performed the readouts. We want to make sure that these effects are not completely transient, but that they will carry for a period of time. We also had some tissues treated with angiotensin II to mimic the hypertrophic myocardium, and also to mimic some conditions of hypertension, because it's known that patients with pre-existing conditions such as hypertrophy or high blood pressure actually do much worse from if they contract COVID. And so what we can see here that in this model, clearly the viral proteins are being produced. You can see Western blotting for viral nucleocapsid protein, significant increase over 48 hours. And what you can also see here is that the number of viral particles that are shed by the tissue, by BioEyer tissue, also increases with time. Working in CL3 facility is a completely different game. People can't really look over the microscope as we normally do with the assessment of BioEyer tissue. You can see the person who's fully gowned in a CL3 facility taking a movie of the moving wire in the BioEyer system using a cell phone camera. So we really had to adapt. The cell phone camera is staying in CL3 facility. We had to adapt our imaging to the new environment. And our ability to do this was really enabled by having this plastic plate that we created by Halin embossing and craft. And so another question is, does this BioEyer tissue have enough machinery to really engage with SARS-CoV? And the answer is yes. If you look at published interactome of SARS-CoV and proteins and phosphoproteins that are expressed in the BioEyer, there is ACE2, the receptor is there, but also about 106 other proteins and 117 phosphoproteins that could interact with SARS-CoV. And so if we then look at these AAV therapeutics, obviously there are a lot of microRNAs that inhibit certain processes in AAVs, and we ask ourselves, what kind of AAVs should we use? And then one natural choice are AAVs derived from cardiomyocytes, because you're trying to treat a cardiac tissue. Another choice are iPS-derived EVs. So either iPS-derived cardiomyocyte EVs or undifferentiated iPS EVs. So you can see from this characterization that we can definitely isolate these EVs. They're there, as confirmed by Western blotting, and as also confirmed by TEM. And what you can see here, we just did an experiment where we applied these EVs to tissues that were infected with model, they're not model, they're actually common cold coronaviruses, NL63, and as well to 290. And what you can see here from NL63, that there was a quite reproducible and robust increase in contractility with time if we apply iPS CVs. And that's why we decided to go forward with the SARS-CoV experiments using these EVs. The question is what's inside, what's in these vesicles, and what you can see here that there are quite a number of microRNAs that can regulate stress response. And for example, you can also see from this KEG pathway that there are microRNAs that could affect, for example, HIPPO pathway, and that's a really important pathway in cardiac development, even cardiomyocyte proliferation that is not a common event, but it still happens. Yeah, and you can see here the most upregulated and downregulated microRNAs in iPSC-derived EVs compared to cardiomyocyte-derived EVs. So this is just characterizing what's inside of these EVs. And so if I move to the next slide, now you can see what the tissues look like when they're infected with common cold coronaviruses, 229E, NL63, different doses of SARS-CoV, as well as treated with extracellular vesicles or angiotensin II. And so one thing that's very striking is that electrical excitability worsens very significantly if we add more SARS-CoV. It really becomes worse, which means this tissue is kind of losing ability to respond to electrical stimulation, which is not good at all. This is at 1 week, at 2 weeks as well. And we can also see that at high doses, the longer we wait, if we go up to 2 weeks, the volume of the tissue starts decreasing. Basically, the tissue is becoming thinner, significantly thinner. And as you will see later, that's because we're losing cells from the actual tissue. The common cold coronaviruses really do not affect contractile force appreciably. You can see just transient, and this is really as expected because common cold coronaviruses don't have tropism for the heart. There is some transient loss in contractility after 8 hours after infection. This is just due to handling because there isn't anything there being expressed to cause this. And you can see the force is completely regained after a period of time. And however, if we now go to and look at force traces of SARS-CoV infected tissues, if you look at here, MOI5, and the traces here, MOI5, with time, we absolutely lose the ability of these tissues to beat. That can be partly rescued with application of EVs. If you quantify the number of tissues that are beating after 1 week, it's really only 20% of them are beating in MOI5. This can be rescued by application of EVs. And the same is also true at 2 weeks, quite a lot, significantly lower percentage of tissue beating and much, much lower contractile forces with time. You can see an example here as well. This can be partly rescued, as I said, with EVs. And if we apply EV treatment, we can have about 60% of tissues beating at the end, actually 80% beating at the end of 2 weeks. So it's not 100% rescue, but 80% is still pretty good. Unfortunately, if we look at calcium transients, we are not able to profoundly rescue those. We see significant deterioration of calcium transients and a very small percentage of actually this tissue area beating with time after EV infection, and this cannot be rescued by extracellular vesicles. What's happening inside of the tissue, as you can see from these immunostains, is that the tissues are actually losing cardiomyocytes and the contractile apparatus is becoming completely disorganized. And you can see it's very obvious at MOI5. We can rescue the presence of the cells with addition of EVs. So that's actually quite encouraging. And as you can also see here, basically the number of tunnel positive cells is slightly but significantly lower with EVs. The number of tunnel positive cells indicating apoptosis goes up significantly in the presence of SARS-CoV. If you look on the inside of the cell now with TM, you can see that the normal control tissues look pretty normal. You can see a cell nucleus here, cytoplasm surrounding the cell, and same thing if you infect with a common cold coronavirus such as NL63. However, when you look inside the cell, a cardiomyocyte in this biovirus tissue that was infected with SARS-CoV, things look like there was an explosion that happened on the inside. The vacuoles, there are apoptotic nuclei that are breaking apart, and only remnants of sarcomeres that you can see here. So these cells are quite destroyed to the point that we were not even sure that these were cardiomyocytes when we first got these TMs, but then we were clearly able to pick up some of the sarcomeres. We can see incubation and basically shedding of these vesicles with viral particles and shedding of the viral particles from the cell as well. That's detectable by TEM. The question is about EVs. There was some positive effect on contractility, as well as on the number of cells in the tissue and what's happening on the, how is this happening. We looked at gene expression, and you can see that uninfected tissues and infected tissues cluster very separately on principal component analysis. EV-treated tissues also cluster by themselves. One of the most inhibited pathways from SARS-CoV infection was oxidative phosphorylation. The cells are really losing ability to metabolize properly, and they're moving to glycolytic metabolisms. Obviously, one of the top 20 most activated pathways was coronavirus pathogenesis pathway, and many other pathways that are related to, for example, increasing endothelial permeability, VEGF signaling, cardiac hypertrophy signaling was also quite significantly upregulated. And we also see something like fibrotic signaling as well that was taking place upon SARS-CoV infection. When we add EVs, for example, glycogen degradation was one of the things that was really being turned on, again, pointing to the importance of trying to regulate and rescue metabolism with coronavirus infection in order to preserve cardiac function. And just in summary, I would emphasize that there is a profound cardiac cell death and contractile dysfunction with SARS-CoV infection, that iPSC EVs were able to promote cell survival. And we are also currently carrying out the studies looking at how vascularized cardiac model behaves under infection. This is important because we want to be able to perfuse monocytes. And just from a few preliminary experiments that we've done, we observe a similar trend of profound, even more profound loss in contractile function in a vascularized cardiac muscle that's perfused with monocytes, as well as the ability of EVs to rescue this response as well. At the end, I'd like to thank everyone who contributed to this work. I also like to add that, thanking our funding sources that generally support research in our lab. And in addition, in particular, the SARS-CoV work that I just showed you was carried out with support of ISI and Connaught Fund as well at the University of Toronto. And happy to take your questions at the end.
Wonderful. Thank you so much, Milica. Certainly very timely and important research. So thank you for sharing that with us today. So we'll hold the questions until the end. And I now invite Jean-Christophe to invite the next speaker.
Thank you, Christine. So I'm happy to welcome Klaus Eyer, who is a former graduate from ETH. After his PhD at ETH, he went to Paris for a postdoc, and then he came back, I think 2 years ago, as assistant professor at our institute. So Klaus' research focused on developing novel technologies to measure the functionality on at the individual cell level during an immune response. And the title of his talk is From Bedside to Bench and Back: The Value of Simple Model Systems and High-Resolution Single-Cell Analysis. So Klaus, the virtual floor is yours, and thank you.
Thank you very much for this very nice introduction, and also from my side, thank you very much for this opportunity to present today one of our research stories. So as Jean-Christophe already pointed out, I would like to show you one specific example. So from bedside to bench and back, and how we value simple model systems and how we value our individual cell analysis. Well, maybe first to take a little step back, if it works. Yes. So what we are, one of our research interests is vaccine-mediated protection. And I became interested in this field in 2015 when I was a postdoc in Paris still. And what seems obvious is that vaccine technology has changed tremendously over the last years, and that's even specifically true for the last 7 years, where we have new technologies and new ways to design our vaccines. But the way we measure the vaccine or we measure protection always more or less stayed the same. So usually what we would take a small sample of blood, and then we would analyze whether these antibodies actually bind our vaccine or not. Whereas it's certainly true that a protective vaccine has to have a high titer of specific antibodies. So for example, against SARS-CoV-2, a high titer doesn't necessarily mean that we have a protective vaccine. You can imagine this a little bit like that. You can have a lot of different antibodies against the disease, but if you don't have the ones which are actually functionally the ones which can neutralize the virus, you will have a problem, or you might have a problem. So in 2015, when I was working on this problem, we started to think about how could we change this? How could we change the way we measure it? And one way to try to do it is not to look in the serum, but to analyze each cell individually. So in this regard, antibody-producing cells are very nice model systems in microfluidics and single-cell analysis, because each cell expresses only one antibody at a given time. So if we manage to analyze each cell individually, we also manage to analyze all antibodies as monoclonals, so as individual species. It's also interesting because there are specific cells, plasma cells and plasmablasts, that secrete these antibodies, but they have different lifespans. So you have subpopulations and raw cells, which also allows you then, by analyzing each cell individually, to have a much better resolution, to have much better understanding of the mechanism of what's going on. And lastly, if you look in the serum, for example, we have around 100 different specific antibodies in a serum after a vaccination, or roughly 5,000 in total. So there's a huge variability that we can basically exploit again by measuring monoclonal antibodies. And in a way, what we do a little bit different is usually people would then turn to single cell sequencing and look at the sequences and try to compare them and try to have different results, or to have evolutions over time. So what we are interested in is the functionality, actually. So how much antibody is produced by an individual cell, and whether that antibody actually has an affinity towards a certain protein or not. So in a way, what we want to do is we want to link this functionality, but that also requires us to work with living or active cells. So we cannot take cells from dead tissue, or we cannot take dead cells, because they need to do something in our assays. We also would like to be quantitative and fast, so to correlate our ex vivo analysis for the moment to the in vivo situation. So usually we try to aim to be in the minutes to hours range. Also, functionalities can change over time, and we would like to capture this change. So therefore, what we decided is to have more than one measurement point. And last but not least, you not only want to analyze one individual cell, you want to have a high throughput and the ability to work with low cell numbers. So somewhere in the range between 1,000 and 1,000,000 cells. And the solution we turned into is droplet microfluidics. So in short, what we do is we basically have our cell suspensions. For the presentation today, these are going to be mostly splenocytes with antibody secreting cells, but it can be almost any different or any tissue which is homogenized and suspended. And we basically encapsulate these using droplet microfluidics into small containers. You can see here one cell within one small compartment. But what we do differently than the literature, usually we would take these droplets and screen them like in flow cytometry one by one. But because we want to have this analysis over time, we basically immobilize them between two glass slides in a small chamber. So we can basically measure each individual droplet multiple times. So we get an idea how the functionality develops and how different functionalities or different degrees of activity also change over time. And this is what you can see here roughly in this scheme. So these are individual droplets. And in each droplet, we have this line. So these are magnetic nanoparticles that we use as an active surface to perform immunoassays. We have the cells shown here or here. And basically, we just test whether the cell secretes a certain protein or not. So in the case of today, we're looking at, for example, IgG secretion, so antibody secretion. And if the cell secretes IgG, you see this line appearing, and we can analyze this individual droplet afterwards. It's important also to stress that you don't have to basically use antibody secreting cells, or you don't have to look at antibodies. The whole system is based on immunoassays. And the nice thing about immunoassays, if you change the different antibodies, you can change the analyte. And I'm also happy to share with you that we have recently translated one of these systems where we look at circulating tumor cells into clinical trials together with our collaborators in Montpellier. But for the sake of today, we will stay with the antibody secretion. So what we started to do is we basically wanted to have an idea about the antibody repertoire that ensues vaccination. So in terms of numbers that we can extract, we can have an idea about the quantity. So you can have a look how many cells secrete IgG. You can have an idea about the secretion rate, so how much each cell individually is producing, but also you can make statements about the quality, whether it has a certain affinity against a certain protein or not. And also in this regard, also the specificity. So we have used this multiple times now to analyze basically the antibody repertoire after immunization. But if you go back to the first slide, to the first thing that I presented to you, that basically antibody titer is not representative of protection, that's also true for this measurement here. So what we are trying to do, or what we started in 2019 when I started at ETH, is we're trying to develop assays that allow us to measure secondary antibody functionalities on the single antibody assay. So for example, neutralization, antibody-dependent cellular cytotoxicity, complement cytotoxicity, or opsonophagocytosis. And this is also the starting point of the story that I would like to share with you today. So in the beginning, we wanted to develop an assay, and we also wanted to have proof of concept experiments in an animal model to basically study whether our assay is working properly and gives reliable results. So I'm going to show you one example, staying with the topic from the former speaker with SARS-CoV-2, where we're interested in measuring neutralizing antibodies. And this is mainly the work of Ingebjørg Sigvalladottir, which is a PhD student in my lab. So in June 2020, we wanted to have, we started with this development of this neutralization assay. And what we decided relatively early on is that we will focus not only on neutralizing antibodies, but we basically only look at competing antibodies that compete with the cell receptor and for infection. So in a way, so here again, you have the virus with the spike protein, and this spike protein binds to the ACE2 using a small subunit, which is called RBD. And on this RBD, which is shown here, we basically have 4 major epitopes that we can bind to with antibodies. And usually class 1 and class 2 are neutralizing antibodies, so they compete with the ACE2 binding, whereas class 3 and class 4 are not. So at the end, what we wanted to have is an assay that allows us to measure whether our antibody competes with the ACE2 binding. And this we have developed over the last months or the last years. So basically what we have again, we have our magnetic nanoparticles that form these beads that you might remember from before, which we have a capture agent. So for example, for our mouse studies, we have a kappa light chain VHH, which basically binds all antibodies that are produced. We also have a detection reagent, which is labeled with 647. And then we also have our antigen, which is in this case the RBD, which we detect with a 488, so in green. And we also have ACE2, which we detect at 750. So in the case, for example, you have a cell here, which produces a neutralizing antibody. You will have signal for the IgG. We will have signal for the RBD, but we don't have any signal for the ACE2. In the case we have, for example, a binding antibody, you will have a signal for the IgG, signal for the RBD, which you can see here, but also signal for the ACE2, which you can see here. And then, for example, we can also have non-specific ones and so on, but you can basically discriminate all these different antibodies. And the nice thing about the system is that you can not only say whether it's neutralizing or not, but you can also get some quantitative numbers. So you can basically measure the apparent affinity of each individual antibody. And we have calibrated this with around 8 antibodies for this system, and they're all more or less are equal or show equal affinities as to SPR. What is also nice then afterwards, we designed this competition score where we basically measure the affinity against RBD and the affinity against ACE2 and divide one by the other. So if you have, for example, an antibody which binds, both of them should be the same. So we have basically a value between 0 and 5. And the higher this number gets, the higher the difference between RBD and ACE2 binding is, and the better the antibody neutralizes this specific target. And you can basically see this here where we have measured with 8 commercially available antibodies from different classes. In green are shown the neutralizing antibodies. For example, for this class 2, which has an affinity of 12 and a competing of 85, so it's neutralizing. For the others, for example, a class 3 antibody which does neutralize, we don't get any number above 5. You can also see that we are not able to find all neutralizing antibodies in this specific case. It's a curious case of an antibody with a very low affinity towards the RBD, but somehow it's neutralizing. But for example, this would be missed in our analysis. But nonetheless, what we tried to do next is once we had this assay characterized, and there's a lot more controls, but I'm not going to show you. We wanted to have some proof of concept study. We wanted to know how it actually behaves with real cells, with cells from an in vivo model. So what we're trying to do is that we immunized mice using RBD with an added neutralizing antibody. And the idea was we would use a concept which is quite well known and quite described called epitope masking. So basically by adding this antibody, we mask this specific epitope where the antibody binds from the immune response. And therefore we should have, or we should shift the immune response or the antibodies away from said epitope. And that has been described quite well by Professor Heimann in Uppsala and her group. But we wanted to have a little bit more quantification to really understand what is actually the impact on the quantity and the quality of these neutralizing antibodies. So first thing we can do is we can, of course, go back and have a look at the serum. So we can see here, for example, that the serum blocking potential, so measurement of neutralizing antibodies, decreases with the amount of neutralizing antibodies that we add, which of course is something that we have expected. At the same time, we can also have a look at the frequency of these cells that secrete these antibodies, and they are more or less similar. So there's no significant change between the 3 conditions. So we know that the number of cells is more or less the same in all 3 immunizations. The last point, what we can see is we can measure the affinity against RBD, and there we can see the first difference. So if we don't have any masking agent, we get a specific affinity of around 8, and this gradually, or the median affinity, sorry, and this gradually decreases the more you mask the antigen, which also seems logical. It's a small protein. We take away the most immunodominant epitope, so therefore we should have less antibodies that bind specifically, or we should have less binding of these antibodies. What's interesting to look at, if we basically look at specifically this population of competing antibodies, so the ones that are actually neutralizing, you can see that even if we mask a little bit, we start to see significant decreases in the number of these cells. So even if we don't have anything on the population level of all 4 classes, if you just look at the ones that compete, we decrease the frequency significantly when we mask more and more. And also individual competitive scores, they significantly decrease. So that means we have less antibodies or less cells that secrete competing antibodies, neutralizing antibodies, and also each antibody by itself neutralizes less on average, which also shows you the exact, basically what we also expected from this model system where we basically block one epitope. So in parallel, collaborators from ours in Germany, Professor De La Rosa at the MDC and Charité, have been working on a different problem. So they were trying to have an assay which allows them to measure soluble ACE2 in serum of SARS-CoV patients. And what they have seen over time is that basically, once they've established the assay to measure this, that the soluble ACE2 correlates with disease severity. That's something you can see here. So on the y-axis, we have the amount of ACE2, which you can find in the serum for healthy donors, WHO status 1, 2, and so on. And for severe disease, we get, or they got regularly the highest number of soluble ACE2. And what they also found is that increased concentrations of soluble ACE2 correlated very well with decreased concentration of neutralizing antibodies. But the problem is again here, because you have a titer measurements, you have soluble ACE2, you have antibodies, you have different people, you have different genotypes or different backgrounds and so on. We don't, or they were not sure whether it's actually a correlation or whether there's a causal link between the two. In a way, what we can do then afterwards is basically we apply exactly our same assay to measure neutralizing antibodies, and we mimic basically these different conditions in mice. So we would immunize them with different masking agents, and in this case, we don't need neutralizing antibody anymore, but we would use soluble ACE2 to mask the epitope to see whether there's actually can be also masked by this endogenous protein. And what's quite interesting to see is that indeed that's true. So here you can see again the same plot from the titer measurements from before. So we have in orange, we have, or sorry, in gray, we have 0%, so just RBD immunization. And the more we mask, the more this curve shifts to the left. And that speaks basically to having less and less neutralizing antibodies. The same thing you can see here in the same representation as before. So we basically, the more we add, we have less and less antibodies that are actually able to block this. If we look, or if we compare, for example, neutralizing antibody and soluble ACE2, both are behaving more or less the same. So here again, we have the frequency of B cells that secrete these competing antibodies, so neutralizing antibodies. So if we don't add anything else, basically we have around 17 to 18% of cells that actually secrete competing antibodies. And this dramatically decreases if we mask epitope using neutralizing antibody. But the same is true if we mask epitope with soluble ACE2. So basically, if we only mask a little as a control experiment, we don't see any significant difference. But if you increase the masking, you also increase or decrease the number of cells in that regard. And both of them follow the same dose responsive trend, which was quite nice. You might also ask, well, basically in a patients, we don't have RBD, but we would have spike protein, but the same holds true for spike as well. So we see this when we mask the protein, we basically decrease the frequency of these cells that actually produce neutralizing antibodies. So altogether, that was a short summary of that study, but altogether that indicates that the presence of a high concentration of soluble ACE2 in serum basically could lead to the decreased concentration of neutralizing antibody by masking this specific epitope, which is so important to produce these antibodies, and therefore allows the virus to infect the cells and therefore leads to severe disease in that regard. So with this, I hope I've given you a short overview of this bench to or bedside to bench and going back. So the last question I would like to address is, so what are we going to do with these results in the future? So on one hand, we also investigating the influence of this masking in different diseases and different applications. So some of our projects are together with the group in Berlin, and we have also other collaborators which are interested in. We would also like to have basically to find these antibodies with the highest neutralizing potential and basically get access to the sequence. And that's the work of one of my PhD students, Kevin, which basically works on sorting these functional cells by linking the phenotype and genotype together. And lastly, this small project about neutralization, which I've shown you, is also part of a larger study where we want to quantify the quality of antibody-mediated protection and also want to aim towards predicting the duration of protection together with other measurements such as metabolic flux on the individual cell level. And with this, I'm at the end, and I thank you very much for your attention, but I also would like to thank all the people that have been involved in these studies and have worked hard over the last 2 years here at ETH, and I'm happy to take any questions.
Thank you very much, Klaus. I guess we are now at the Q&A session. So I don't know, can we, so we should be able to see any questions, huh? Correct?
Should, yeah. So maybe we can open it up, and then I just invite everyone to please share your questions with us in the Q&A panel. But wow, these are just fantastic talks, and we certainly see the tremendous potential and impact of these in vitro models. As someone who doesn't use these models, but and relies on animal studies, I just wanted to get a sense, for the extent to which you feel these models do provide definitive data. I mean, data that you feel good enough about that you would then perhaps push a new therapeutic or diagnostic approach into the clinic. Maybe Milica, I looked at, with your data, on the iPSC EVs. I mean, the data that you've obtained on those in your in vitro model, is that something that's definitive enough, that's exciting enough, that you think that it could be pushed into the clinic now, or would you then have to go and perform these types of studies in vivo? And she may not be with us. I think
she's muted, no? Or she
may be muted, yeah. Milica, can you hear us? Maybe the same question for Cornelia. In your great models, the in vitro models, the corneal culture models, for example, size matters and some of the different relationships that you're finding. Would you then feel that you have to go do an animal study, or is it sufficient to have this in vitro model, or maybe the in vitro model better recapitulates the human situation, given that we're largely working in rodent models if we're using animals?
I mean, I think the question is valid in my case particularly, because I think a rodent cornea does not really reflect so much the human situation. I mean, it's not going to have the same thickness of the epidermis is just, I mean, the layer is just much thinner. So the penetration question is, of course, an issue. On the other hand, I'm also wondering, and I don't know the solution, and neither do my collaborators, whether the penetration issue is so much of an issue, because of course, if you have done a transplantation, you have already created a hole, I mean, and at the place where this is now growing together again, and particularly if you're just treating during the time of when the wound is healing, penetration might anyway, again, be very much different as if it was a completely intact cornea. I mean, I think we have to test out many more models. At the moment, we are just on these in vitro models that I mentioned, but also with the animal corneas. I mean, there you also have some supply that you could get from slaughterhouses, which would not really, put up the statistics for the animals experiments in that case. So
okay, thank you.
Maybe I have a follow-up question. So Cornelia, so when we apply drugs topically, I mean, clearance is very quick, huh? I mean, because of the tears. And so, I mean, I guess, how much do you need the compound to stay on the cornea, before, or before you see some effect? So are you able to, you show that size was important in terms of penetration, but then in terms of retention, what I mean, do you have any idea on how you would plan to apply the formulation? Or
I mean, to my knowledge, also the approved the approved product, and also the single chain TNF alpha blocking antibody are applied not in terms of like a depot that is releasing it continuously, but really as an eye drop. And it's, I mean, it's still just putting it on and holding close your tear channel to keep it there for some time. But I agree with you that this is not going to stay there for endless time. I was very therefore that's why I brought it in, surprised by these bevacizumab studies, where also in some of them they did not plug any outflow route, but really just gave it on top, and then nevertheless, with like 4 applications a day, they saw an effect in the neovascularization. But also there, apparently, if the cornea is irritated, the barrier might not be as intact as you expect, or then they give, there are some of these also in normal eye drops, some of these compounds which affect the penetration of the epithelium, benzalkonium, something is is used, but of course you don't want to irritate it too much in a condition where you exactly don't want irritation. So,
but still, it would have to be applied. I mean, as maybe 4 times or more per day. Yeah, still, yeah.
I think so, but
the maybe it's a bit viscous too. Yeah, I would assume.
Yeah, the hope would be that you just have to apply it for a short time period. I mean, that's the same with these with the corticosteroids, which are just applied at the present for 6 months. But I mean, at most, because of the side effects that this treatment can have by itself. So the hope would be that you can taper it off once the inflammation is gone, because if the inflammation is gone and you haven't had new blood vessels forming, the hope would be that you can keep or build up a new privilege, immune privilege.
May I ask another question?
Yes, absolutely.
Okay. No, I have a question for Klaus. So I liked a lot your work about the soluble ACE and ACE2, and I was wondering, would it make sense before we vaccinate people to deplete soluble ACE and then inject the vaccine, or that would has that absolutely no sense?
It would make sense, right? But it's a very hard thing to do, and I might not be the strategy you would like to choose to do that. I don't know how much I could say about the rest, but there's different.
Would it be dangerous to do that? I mean, to inject a high amount of antibody just prior to injection, or do you expect that it could create any problems? Or,
I mean, you could run the risk of having some immune complexes, which you don't want, I guess. I mean, there's definitely ways you could. So there's definitely ways you could design it to have as a therapy, or not a therapy, but basically to have this antibody injection to get rid of the ACE2. But it's quite a lot. I mean, what I've shown you is just the active ACE2, and 99% of it is actually inactive. So it can go up to micrograms per milliliter. So you would need quite a lot of antibodies in that regard.
I see.
Klaus, why does the soluble ACE2 not affect infection of the virus?
It's a good question. I mean, there's also some studies around where they show that the virus itself has actually has ACE2 on the surface of it. My hypothesis, or the way I work with it, is to think about that in a way you have a one-to-one interaction. So if you have, for example, the soluble ACE2 on the virus surface, it's a one-to-one interaction. But if you get into contact with a cell, at some point you will bind to it, and then you will have to start avidity effects, right? So it's still much more efficient for the virus to bind to the cell.
But you could argue the same way with the antibody, which is only bivalent, whereas you don't have any patching interactions, you know.
But for the antibody, it needs to be in a specific distance, right, for it to have a bivalent interaction. And it might be that the spike proteins are too far away from each other. I mean, I made a back on the envelope calculation. I'm not 100% comfortable with it, but it seems that they are further away, that only one antibody can only bind to one spike protein. Yeah. Whereas for a cell surface where you have the diffusion and everything, you can have avidity. I'm also open for any other ideas.
I think we have a question actually here from Michael Sefton. So I'll just read this through. Cornelia, this is for you. Do you think the insights gained for DC migration in the cornea transplants could be relevant to transplants in vascularized sub-Q sites?
So for other organs, for example, like the skin, or what is meant with vascularized?
Michael, I have
for other vascularized
I
tissues.
think that's probably what he means. Yes.
Yeah, I really don't know, but reading a bit about transplantations, it seems that in certain organs, I think like lung or kidney, it's not so clear if antigen presenting cells only leave via lymphatics, or if they can also somehow miraculously make it into the blood and then priming occurs in another organ, like in the spleen. And somehow in this cornea, it still seems rather a different situation because because of this it's not so highly vascularized situation. And so there it seems more really focused towards this lymph nodes and establishing this lymphatic connection that is very relevant for the priming. I'm not sure how much it applies for the other transplantation setups. There are also some data, lymphatics have another role, which is also the drainage of tissues. And in the lung, for example, there are some studies that would indicate that if you block lymph angiogenesis after a lung transplantation, it's actually bad, because in the very beginning, you want drainage and clearance of the inflammatory, environment. But on the other hand, you also don't want migration of antigen-presenting cells to the lymph nodes. So it's a bit of a dilemma that it has two effects which are to a certain degree counteracting each other. But the edema formed in a tissue like the lung is also caused by the high degree of vascularization, which again in the cornea you would not have to start with. So I'm not so confident it will be translatable to other transplantation setups, unfortunately.
Okay, thank you. Thank you. Maybe I think we've just got a couple minutes left. Klaus, on your first few slides, you talked a little bit about a translated system that was focused on circulating tumor cells, and that was moving into the clinic, I think. Could you tell us a little bit more about that? Sounds very exciting.
Yes, it's also a good example where it didn't work with the simple model system.
Right.
So we were always trying, so because we look at cells one by one of the first applications we thought we could try is to have a look at circulating tumor cells, because you have so few of them and you might be able to spot them if you look at each cell individually. So we try to basically have different assays for different cancer types and different functions that circulating tumor cells have. And then we looked at basically whether we can find these cells. And there, for example, first thing we tried is always to spike in some cancer cell lines into fresh blood, and we're never able to find anything. Until we tried to convince our clinical collaborators to give us one or two samples. And there we can see the cells. And I think just in our case, all the cancer cell lines, they like to stick to things. And because we spiked in like 1 or 10 cells, they were already gone before we even had it in a chip. And now, I mean, we are trying, so now we have basically a study or 2 studies going on with prostate cancers where we want to try to see how many cells you have, because you can use this as a prognostic marker, and you can also use this to follow therapy, because if the therapy works, this number should decrease over time. And it seems to work quite nicely. I mean, we outperform the current system, which is not FDA approved, but cleared by the FDA. But the studies are just started like 2 or 3 months ago, so we'll have to see how it goes.
Very exciting. Wow, and it's such an early stage in your career. That's amazing. That's awesome.
I think we have a hand raised.
Do we? I'm sorry if I missed that. Oh, here we go.
Yeah.
Peji. I'm not sure. Peji, you're welcome to ask a question. I'm not sure if we'll actually be able to hear you. Alternately, you can just type your question in the Q&A panel. So if you go to the bottom of your screen and click on Q&A, you can just type your question there. Christine, are we able to heir the participants?
Yeah, I have, when hands are raised, I have permitted talking.
Okay, perfect.
Just not sure if there is a hand. Yeah.
There we go. The hand was raised and it is gone now.
It's muted now. Okay.
Okay. Jean-Christophe, do you have any other questions?
No, I think I'm just fine. Maybe if there are no more questions, I mean, maybe we can take just a longer break. I mean, we have only a 5-minute break, and then we'll be just a bit more before we resume the conference.
That sounds great. So we'll let everyone take a break now until 10:40. I think that's correct. And, you can see just fantastic talks this morning or this afternoon for some of you. And you can also see the complementarity and synergy between the research that is going on at these two different sites. So lots of opportunities, I think, for collaborations, which will be fantastic. So enjoy your break, and we'll see you back in a few minutes. Thank you.
Thank you.
Thank you very much.
Oops.
So I think we'll get started, everyone. Welcome back. This is our next theme, which is focused on engineering molecular structure for imaging. And our first speaker is Professor Raymond Riley. He is a professor in the Leslie Dan Faculty of Pharmacy at UofT, as well as the director for the Center for Pharmaceutical Oncology. His research is focused on developing novel radiopharmaceutical agents, including the discovery, preclinical development, and clinical translation of these agents for molecular imaging or molecularly targeted radiotherapy of cancer. And the title of his presentation today is Molecular Imaging of the Delivery of Trastuzumab to Brain Metastases in Patients with HER2-Positive Breast Cancer, enhanced by MR-guided focused ultrasound. Welcome, Dr. Riley.
Thank you, Christine. I'll just set up my slides here. Okay, so thanks so much for that introduction. Good morning, everyone. It's my pleasure to describe to you today an exciting and landmark clinical trial that I'm collaborating on at Sunnybrook Health Sciences Center in Toronto. And as Christine mentioned, the trial is aimed at studying the delivery of trastuzumab, a drug used to treat breast cancer, to brain metastases in patients with HER2-positive breast cancer, enhanced by MR-guided focused ultrasound. In this clinical trial, molecular imaging with radio-labeled trastuzumab is being used to assess the delivery of the drug to tumors in the brain. And I think the results of the trial, as I'll show you today, illustrate the power of molecular imaging to evaluate drug delivery. So just a couple of disclosures here. Incytec is the manufacturer of the Exablate MR-guided focused ultrasound system and is a sponsor of the clinical trial. And the radiopharmaceutical indium benzyl DTPA NLS trastuzumab the development of this radiopharmaceutical was sponsored by a grant from the Canadian Cancer Society. And the agent is an investigational agent approved for study in the clinical trial under a no objection letter from Health Canada. So in my presentation today, I will outline the challenges of treating brain metastases from HER2-positive breast cancer and explain how MR-guided focused ultrasound may overcome some of these challenges. I'll then describe the clinical trial at Sunnybrook Health Sciences Center, where we're investigating MR-guided focused ultrasound for enhancing the delivery of trastuzumab to brain metastases in patients. As I mentioned, SPECT-CT imaging molecular imaging is being used to evaluate drug delivery. At the end of my presentation, I'll just briefly mention some future studies in this area of alpha particle radioimmunotherapy combined with MR-guided focused ultrasound for treatment of brain metastases from HER2-positive breast cancer. So about 15 to 20% of breast cancers overexpress the human epidermal growth factor receptor number 2, or HER2, and this was previously a poor prognosis marker due to its role in promoting tumor growth. However, the development of HER2-targeted therapies such as trastuzumab, pertuzumab, and the antibody drug conjugate trastuzumab DM1 have significantly improved patient outcome, even in patients with metastatic disease. However, the brain remains a sanctuary for tumor cells. As many as 1 in 2 patients who progress to metastatic HER2-positive breast cancer ultimately develop brain metastases, and these are very difficult to treat. One of the reasons that it's difficult to treat brain metastases is the blood-brain barrier. So this is a physiological barrier composed of brain endothelial cells, astrocytes, microglia, and a basement membrane, which poses a selectively impermeable barrier to most drugs, and including drugs for treatment of cancer. So the blood-brain barrier limits the delivery of trastuzumab and other HER2-targeted drugs to the brain. In fact, one study showed that trastuzumab concentrations in the CSF in patients were more than 400 times lower than in the blood. So one approach to improving the delivery of trastuzumab to the brain is MR-guided focused ultrasound, which is being studied by our collaborator, Dr. Halin. Start my laser here. Okay. It's being studied by our collaborator, Dr. Halin at Sunnybrook Health Sciences Center. And in this approach, ultrasound energy is precisely applied to the brain to transiently and spatially disrupt the blood-brain barrier at a predetermined location in the brain. The application of MR-guided focused ultrasound disrupts the endothelial cells forming the blood-brain barrier by causing intravenously injected microbubbles, definity microbubbles, to oscillate in the vasculature. The opening of the blood-brain barrier is transient and it returns to normal physiological state within a few hours. So MR-guided focused ultrasound may be a way of improving the delivery of trastuzumab across the blood-brain barrier. During the open phase of the blood-brain barrier, intravenously administered trastuzumab may cross the endothelial barrier forming the blood-brain barrier and reach the brain, where it could then be used to treat brain metastases. This approach was previously shown by Dr. Halin in non-tumor bearing mice. Application of MR-guided focused ultrasound to the brain of these mice disrupted the blood-brain barrier, and this was revealed by T1 weighted MRI with gadolinium DTPA contrast, showing an accumulation of the contrast agent in the area of the brain where the focused ultrasound was applied. Analysis of these images, these MRI images for MRI intensity, showed that with focused ultrasound, the intensity of the contrast signal in the brain was significantly increased compared to without focused ultrasound. An ex vivo analysis of the brain using an ELISA technique to detect the presence of trastuzumab revealed increased uptake of trastuzumab into the brain of mice compared to mice that were not treated with ultrasound or that were treated with a lower acoustic intensity of ultrasound. Molecular imaging technologies such as SPECT or PET offer the opportunity to visualize the delivery of trastuzumab to the brain in humans using radio-labeled forms of the drug. For example, one PET study showed that uptake of zirconium-89 labeled trastuzumab in a brain metastasis in patients due to local disruption of the blood-brain barrier caused by the presence of the tumor. So the clinical trial at Sunnybrook, which I'm collaborating on, is being led by Dr. Nir Lipsman, a neurosurgeon, and Dr. Halin and their clinical team. In this trial, patients with brain metastases from HER2-positive breast cancer are being treated with trastuzumab, pertuzumab, and chemotherapy, taxane chemotherapy. These patients have a baseline study done after injection of a radio-labeled form of trastuzumab, indium-111 benzyl DTPA NLS trastuzumab. This study is intended to evaluate the uptake of the radiopharmaceutical, which is a radiotracer of trastuzumab into metastasis in the brain prior to applying MR-guided focused ultrasound. After injection of the radiopharmaceutical, the patients undergo SPECT-CT imaging at 4 hours and 48 hours. 2 weeks later, the patients then return and have MR-guided focused ultrasound using definity microbubbles and MRI with gadolinium DTPA, as well as trastuzumab administration. As part of this second study, the patients also receive a second injection of the radiopharmaceutical, and imaging is then performed again at 4 hours and 48 hours after injection of the radiopharmaceutical. So the radiopharmaceutical was developed by my research group, and it consists of trastuzumab that's modified with the metal chelator, benzyl DTPA, to bind a radionuclide indium-111. Indium-111 emits gamma photons, which are useful for SPECT imaging. but it also emits low energy Meitner-Auger electrons, which could potentially be used for radioimmunotherapy. The trastuzumab molecule has been further modified with the nuclear localizing signal peptide, which promotes nuclear uptake in breast cancer cells after HER2 binding. And this is intended for future application exploiting the Meitner-Auger electrons from indium-111 for radioimmunotherapy. In the clinical trial, only the imaging properties of the radiopharmaceutical are being utilized, but the radiopharmaceutical, as mentioned, has the potential for radioimmunotherapy as well. So the agent is what's called a theranostic, able to both have diagnostic application as well as radiotherapeutic application. In order to prepare the radiopharmaceutical for patients, we formulated a radiopharmaceutical kit, which is a single vial that can unit dose vial that is manufactured under good manufacturing practice conditions, GMP conditions, that contains 5 milligrams of benzyl DTPA NLS trastuzumab in 1 ml of ammonium acetate buffer pH 5.5, which is suitable for labeling with indium-111, with a patient dose of indium-111. The final radiopharmaceutical is prepared by adding indium-111 directly into the vial. The labeling efficiency is very high, more than 90%, and thus no post-labeling purification is needed. The kits are manufactured in advance and are stable at 4 degrees C for at least 6 months, but the final radiopharmaceutical is prepared individually for each patient on the day of the study. So this table shows the characteristics of 4 patients that were enrolled into the clinical trial. These patients all have HER2 positive breast cancer, and they subsequently developed brain metastases after anywhere from 1 to 8 years post diagnosis here. And these brain metastases in the patients were treated with whole brain radiotherapy or stereotactic radiotherapy. And the patients are receiving trastuzumab and pertuzumab enhanced by MR-guided focused ultrasound in the clinical trial. This slide shows a photo of a patient undergoing the procedure, MR-guided focused ultrasound. You can see the ultrasound helmet surrounding the head of the patient. As mentioned, 2 studies are performed, one at baseline prior to administering MR-guided focused ultrasound, and a follow-up study 2 weeks later when the patient receives the MR-guided focused ultrasound combined with trastuzumab treatment. As mentioned, the patient receives an infusion of therapeutic trastuzumab, but also an injection of the radiopharmaceutical, either at the baseline study or at the 2-week follow-up study. And during the follow-up study, the focused ultrasound is applied. It's about 2-minute procedure, very low intensity ultrasound, 200 kilohertz, applied with the exablate inside focused ultrasound system following infusion of DEFINITY microbubbles. And MRI is performed at 1 hour after the MR-guided focused ultrasound treatment in order to evaluate disruption of the blood-brain barrier. And then again at 18 to 24 hours to determine that the blood-brain barrier has reformed. And SPECT-CT imaging is performed at 4 hours and 48 hours after injection of the radiopharmaceutical. So this slide shows some representative images in one of the patients in the trial, which is patient 1. And the upper series shows a series of images are MRI images, which show that with using gadolinium DTPA contrast, that after the application of the focused ultrasound, there is some leakage of the contrast agent into the brain, which indicates disruption of the blood-brain barrier. However, at 24 hours, the endothelial junctions are reformed and the blood-brain barrier is restored to its normal physiological function. The series of images at the bottom are images either at 4 hours or 48 hours after injection of the radiopharmaceutical in patients receiving MR-guided focused, in this patient receiving MR-guided focused ultrasound. The arrow, white arrow here is actually pointing to the lesion in the brain, And you can see prior to the focused ultrasound at 4 hours, there's very little uptake of the radiopharmaceutical. There's a slight increase at 4 hours after injecting the radiopharmaceutical in this region in the brain. But at 48 hours, we see a significant increase in the uptake of the radiopharmaceutical, comparing the pre-focused ultrasound images to the post-focused ultrasound images. And these images can actually be quantified using what's called a standard uptake value. And so by quantifying the uptake of the radiolabeled trastuzumab into this region of the brain, it can be shown that there's a significant increase in the uptake of the radiopharmaceutical into the area of the brain that received the focused ultrasound, and also corresponding to the lesion in the patient. This slide actually shows the image analysis for all of the patients and all of the lesions that underwent focused ultrasound. It's showing the increase in the uptake of the radiopharmaceutical post-focused ultrasound compared to pre-focused ultrasound at the baseline study. And you can see in each case, there's a significant increase in the uptake of the radiopharmaceutical into the sonicated area of the brain. However, image analysis of regions of the brain in 2 patients here that did not receive focused ultrasound shows no significant change in the uptake of the radiopharmaceutical in that region of the brain. The slide actually maps the increase in the uptake of the radiopharmaceutical into areas of the brain that received focused ultrasound with the MRI images. And you can see the scales here anywhere from 0 to 300% or 400% increase in uptake of the radiopharmaceutical overlaid with the MRI scan to show the region that was also sonicated. And you can see that there's in each case for these 2 patients in different regions of the brain that underwent focused ultrasound, that there's a significant increase in the uptake of the radiopharmaceutical, up to 2 to 4-fold compared to the baseline imaging studies that were performed. This is an example of an MRI scan of a region in the brain of a patient that did not receive focused ultrasound, and you can see that there's very little change in the uptake of the radiopharmaceutical in this patient. So the MR-guided focused ultrasound combined with the molecular imaging is actually performed on the first of 6 cycles of therapy that these patients are receiving. So the patients are receiving therapy every 3 to 4 weeks with trastuzumab and pertuzumab and taxane chemotherapy. And the MR-guided focused ultrasound combined with the imaging is only performed on the first cycle. So these patients are continuing to be monitored in terms of their changes in tumor size in the brain. And this shows some interim results from monitoring changes in tumor size. And you can see that in all patients here, or at least these 4 patients that were evaluated, there's a decrease in tumor size over time in these patients who have received MR-guided focused ultrasound combined with trastuzumab and pertuzumab. So based on the exciting results of this clinical trial, we're now thinking about what could be the next step in this approach. So one approach may be to develop a radioimmunotherapeutic analog of trastuzumab that could be combined with MR-guided focused ultrasound, because previous studies in our lab actually showed that indium-111 labeled trastuzumab was at least 10-fold more cytotoxic than trastuzumab in vitro for killing breast cancer cells, and was more effective for treatment of HER2-positive tumors in mice than trastuzumab alone. So we're now developing a radio-labeled form of the drug that could be used for radioimmunotherapy. So this is trastuzumab modified with a metal chelator DOTA that can bind either indium-111 for Meitner-Auger electron radioimmunotherapy, or actinium-225, which is a very powerful alpha particle emitter that has received a lot of attention recently. And so the alpha particles emitted by actinium-225 or the Auger electrons emitted by indium-111 have very high linear energy transfer, which makes them highly damaging to DNA and lethal to cancer cells. So that there is the potential here to significantly improve the therapeutic outcome by combining radioimmunotherapy with MR-guided focused MR-guided focused ultrasound enhanced brain penetration. Some early results in our work to develop a radioimmunotherapeutic analog are shown on this slide, which is work done by Misaki Kondo, a PhD student in my research group. And this slide actually shows that actinium-225 labeled trastuzumab is very powerful at reducing the clonogenic survival of 2 different breast cancer cells that are HER2 positive. In fact, it reduces their clonogenic survival in culture to less than 0.1% at very, very low nanomolar concentrations. And we found actually comparing indium and actinium, that actinium DOTA trastuzumab was more than 1,000-fold more powerful than indium at decreasing the surviving fraction of these cells. And as I mentioned, indium itself was 10 times more effective than trastuzumab for killing these cells. So just in conclusion, I hopefully have shown you that MR-guided focused ultrasound can enhance the uptake of indium-111 labeled trastuzumab into areas of the brain that are sonicated by transiently disrupting the blood-brain barrier revealed by MRI, and the increased uptake of the radiopharmaceutical was further revealed by SPECT-CT molecular imaging. Interim treatment response monitoring reveals decreased tumor size in the brain in patients treated with this interventional technique, MR-guided focused ultrasound combined with trastuzumab and pertuzumab. And in the future, we plan to develop the radioimmunotherapy with trastuzumab labeled with the powerful alpha particle emitter actinium-225, combined with enhancing the uptake of the radioimmunotherapeutic agent into the brain as a potential even more promising strategy to amplify the therapeutic response. So I just want to again acknowledge the clinical research team at Sunnybrook that I have the privilege of collaborating with, that led by Dr. Nir Lipsman and Dr. Kalervo Haninen. And I also want to acknowledge the individuals in my research group who helped develop the radiopharmaceutical, Dr. Conrad Chan and Zhongli Kai, they both prepare the radiopharmaceutical as well for patients on the day that they're booked for the study. And this study actually was launched right in the middle of COVID-19. So I'm actually quite proud that we were able to launch this clinical trial in the midst of everything else that was going on in the world. And also want to acknowledge Misaki Kondo is a talented PhD student in my group who's going to be continuing with this work to study alpha particle radioimmunotherapy combined with MR-guided focused ultrasound. And the work is sponsored by the Canadian Cancer Society as well as CIHR, Canadian Institute for Health Research here in Canada. And I'm happy to take any questions during the break, and that's my contact information. Thank you very much.
Wonderful. Thank you so much, Dr. Riley. What a great translational research success story. I can tell you that Dr. Riley also has a clean room that is just around the corner on my floor here that he received funding to build. And so he's able to make these imaging agents and radiopharmaceuticals just right here in the building. What a wonderful experience for trainees to gain that experimental exposure and so on. So thank you so much, Ray. And we will move on to our next speaker now.
Yes, thank you, Christine. So the next speaker is Professor Roger Schibli, who is a professor at the Institute of Pharmaceutical Sciences at ETH Zurich, but he is also the head of the Center for Radiopharmaceutical Sciences. It's a joint endeavor between the ETH Zurich, the Paul Scherrer Institute, and the University Hospital in Zurich. Professor Schibli's research covers targeted tumor diagnosis and therapy using radio-labeled compounds. His group emphasizes the biological and pharmacological characterization and optimization of the radioactive compounds, including non-invasive PET and SPET imaging. He will be presenting nuclear diagnostic from bench to bedside. So Roger, the floor is yours. We'll just have to stop sharing from the previous presentation.
I'm having some technical difficulties trying to stop sharing here.
No problem.
Okay. So maybe I can stop it myself. No, I don't know. No, I just cannot. Can someone?
I apologize for this, but for some
reason,
problem.
that's okay.
If you
Christine?
exit Ray, you may have to exit Ray and come back in.
Yeah, I'm just
Yeah,
trying
maybe
to
that would be the way.
even exit. My computer is kind of frozen for some reason. So, oh, there we go.
There we
Okay,
go.
that goes. Okay, thank you.
Okay, thank you very much.
Right.
Jean-Christophe, you can see my slides? Perfect.
Yes.
Just give me see, you can see also the pointer, huh? So thanks very much. Actually, I also have to thank my previous speaker for introducing the topic quite nicely. So I will continue with giving you a bit of insight on radiopharmaceutical development done and performed here at ETH in Zurich, but also at the Paul Scherrer Institute. So, well, we have heard about radionuclides, and radiopharmaceuticals are a bit like the prototypes of so-called diagnostic agents, because they really combine the features of a diagnostics and also a therapeutic agent in one and the same structure. So we have various isotopes which can be used for imaging, and we have isotopes which can be used for therapy based on their physical decay properties. So mainly those we used for imaging are emitting so-called gamma rays, and those which are used for therapy emit particles like Auger electrons, which we have heard, or alpha particles, or beta minus particles. You probably know, I mean, that's since the 50s, we have this, let's say, prototype of application of radioisotopes when it comes to thyroid imaging and thyroid therapy with iodine, isotopes of iodine like iodine 123 and iodine 131 for therapy. Another feature which is interesting for radiopharmaceutical development, and this is actually one of the reasons this type of research can still be done in an academic environment, is that we really are in the so-called microdosing regime. So what we apply in amounts of non-radioactive substances, for example, a peptide or an antibody, is really very, very low. So we are in the nanogram to microgram of cold material we can inject or we inject into patients. That means we can perform reduced toxicity studies for such compounds. So usually it's a single dose toxicity, acute toxicity study in 1 or 2 species. And so this is still affordable, or can still be paid in an academic, from an academic budget, for example. So that is really also one of the advantages. So we do not expect any pharmacological effects of the drug, of the compound we're injecting. So the diagnostic feature or the therapeutic features really come only from the isotopes. And I mean, one of the success stories is really shown here, and what I point out here about this theragnostic approach with this peptide, this somatostatin analog. You see the structure here, which is functionalized with this chelating system we have also seen before. And by simply switching from a diagnostic isotope, gallium-68, which is a PET isotope, to a therapeutic isotope, and then back to a diagnostic isotope, you can really nicely see the differences in these patients. These are patients, studies done at the University of Zurich. So you see the metastasis of these patients suffering from the neuroendocrine tumor before the therapy with lutetium 177, and after the therapy, where you see virtually the disappearance of all these metastases. This type of compounds or applications are actually for many, many years known and have been applied in hospitals quite successfully. For example, the University Hospital in Basel was very famous. Also, the University Hospital in Rotterdam was very famous for its applications. But they never really made it through, a commercial success. Because they were really done in an academic environment at hospitals, on-site production, on-site application. So it was not really, there was never really a phase 3 clinical study performed up to very recently. And this was actually done in 2017 when the NETA trial, the results of the NETA trial came out. Clearly one could see that in these mid-gut neuroendocrine tumor patients, the progressive survival is significantly enhanced in those treated patients, and also the overall survival was definitely increased compared to the controls. This NETA trial, the result of the NETA trial, then actually brought this field to the attention of Big Pharma, and Novartis licensed this compound just briefly after this phase 3 clinical trial has been published. A second success story, also, of course, this was not done in our laboratories, but just to see you or guide you a bit or bring you into the field, is the success story with these PSMA targeting molecules. PSMA is prostate-specific membrane antigen. And again, this compound here, the targeting moiety, the binding site is actually only this diglutamate with a urea bridge in between. That's the binding site, and all this is spacer entity, and also again here the chelating system. Again here you do a therapeutic imaging of these patients. You can see the lesions here, the metastasis in these patients. Then you switch to a therapeutic isotopes using the very same structure here, and then back actually you go to the diagnostic imaging, and you can clearly see that the disappearance of the metastasis in these patients. And one has to point out that all these patients have been heavily pretreated, so they're castration refractory patients. So they have hormone therapy behind them. So that's really the last resort of therapy they can be offered. And also these are actually studies performed in Heidelberg. So where they developed this compound, they're also using actinium-225, the isotope we also have heard before, for alpha therapy. And also there the results are quite amazing. Again, here, a phase 3 study, which was just recently terminated, this VISION trial came out very recently. And again, here, a significant reduced risk of death after the therapy with lutetium, and a significant reduced risk of progression after therapy of these patients. As I pointed out, very last options for these patients to get a treatment. Again, this was licensed by Novartis very recently, and now they are really doing the large-scale multi-center clinical trials, including also in Switzerland. So switching now to our own research, we are focusing for many, many years on another targeting molecule and another target, which is the folate receptor and the folic acid as a targeting molecule. Folate receptor has been reported to be frequently overexpressed in different types of cancer, namely ovarian cancer and also lung cancer. And folic acid is a small molecule which allows, due to its structure and due to its binding to the folate receptor, to be functionalized quite easily at this 2-carboxylic acid. We were not the first ones working with folic acid delivered compounds, including radiopharmaceuticals. But nevertheless, if you look in the literature, what the success stories of this folate receptor alpha targeting drugs is quite limited. There was a study which used the folic acid with a vinca alkaloid as a chemotherapeutic. This was also terminated in 2014. There was an antibody targeting the folate receptor alpha, which also was not so successful. And lately also ADC, an antibody drug conjugate targeting the folate receptor, did also not reach its primary endpoints. So the question arises is what went wrong? And maybe looking at these results of this PROCEED trial, this is again this vinca alkaloid attached to folic acid via spacer entities. And if you look actually, if you would pre-select the patients which have a high expression, so large, high expression of the folate receptor, you clearly can see there is an advantage of these patients compared to those with a medium to low expression, or those which have no expression. So it would be very important to have a pre-therapeutic imaging to maybe select those patients which would benefit from the therapy. Endocyte, which was actually developed, the chemotherapeutic, also co-developed a radiopharmaceutical labeled with this SPECT isotope, this technetium-99, and they performed also pre-therapeutic images. Still, the compound or the therapeutic indication was not so successful, and we think it's mainly because single photon emission tomography is really not so sensitive and not so quantifiable as, for example, PET radiopharmaceuticals. That brought us to the idea that we could eventually develop a folate receptor targeting molecules labeled with this F18, so fluorine 18, which is a PET isotope, so which allows quantification much easier than, for example, SPECT radiopharmaceutical. We started to do this called a pendant approach, so we attached a prostatic group, which was then labeled with fluorine 18 at this site, so you can see here again the structure of folic acid. We switched then to an internal approach where we introduced the fluor 18 into the structure of folic acid. So this is really virtually the same molecule as the parent compound, except that you introduce a fluoride, whereas it's a hydrogen. And then we switched to a pyridine labeled compound simply because of low radio labeling needs with this second generation compound. That's the preclinical images of those compounds. Clearly, the first generation showed low tumor uptake in this mouse model. We see here the tumors, but you see a large intestinal uptake. Also, gallbladder was highly traces highly up. And of course, if you would use such a compound in a human setting where you were looking for ovarian metastasis, this might be not an ideal situation. So the second generation was actually quite successful. You could nicely see the heterogeneous uptake in this mouse tumor model, low uptake in the other regions, the organs, except in the kidneys, where the folate receptor is also overexpressed. And as I mentioned here, the radiolabeling yield was too low. We could not switch that into a clinical setting. So we switched to the third generation, where you clearly can see again high uptake in the tumor sites, specific uptake is in the kidney, and virtually no uptake in the other organs. So with this Azafol compound, we then switched into a clinical setting. So of course, toxicity study had to be done, extensive. Also, GMP production was set up in our laboratories at ETH in Zurich. So we did a multi-center first-in-man clinical study, which I highly discourage everybody to perform because it's really a pain. But nevertheless, we finally managed to recruit 12 patients, 8 small lung cancer patients and 4 ovarian cancer. And we clearly could see a correlation between immunohistochemistry of in these patients where we have high receptor expression. We could also see high uptake of this radiopharmaceutical, whereas in folate receptor negative samples or patients, we clearly could see no uptake. Actually, here this black spot is actually the tumor which is negative. And you clearly there's no uptake of the radiopharmaceuticals. You see background activity in the liver from normal excretion. That's lung cancer patients. Ovarian cancer patients showed again also uptake in lymph nodes, where we have also seen receptor positive lesions. That's another patient. Lymph node metastasis clearly visible with this new radiopharmaceutical. And so that is actually quite an interesting compound, we thought. One has to point out that this compound, and I maybe switch to the next slide, is actually an oxidized form of folic acid. It's not the natural folic acid or the physiological form of folic acid. And this is not, it does not discriminate between the folate receptor alpha, which is expressed on the tumor cells, and folate receptor beta, which is expressed on activated macrophages. So you cannot discriminate between inflammation and cancer. And through a number of steps, and I would say about another 5 years of development, in parallel to the clinical studies, we now have developed radiopharmaceuticals or folic acid derivatives, which allow not only the imaging, but also therapy. Again, here, the chelating system, which allows the radio labeling. We also introduced albumin binding entity to keep the small molecules longer in circulation and enhance the tumor uptake. And in a second, or in the next step, we actually switched to reduced methyl tetrahydrofolate receptor or folic acid derivatives, where they showed selectivity, at least in literature. Was reported that this is now folate receptor selective. And that's what we have now, where we currently stand. So we have here the uptake of the situation in a mouse with the oxidized form of the compound with labeled with lutetium-177. So we hear tumor uptake, which is not that great. We see again kidney uptake because of the receptor expression there. But we could really see an interesting feature that we have the two isomers, the R and the S isomers of the reduced form, show distinct differences. So the S isomer shows a very high uptake in the tumor, but also very high uptake in the kidney of the mouse, whereas the R enantiomer shows almost exclusive uptake in the tumor, much higher than the oxidized form, and very low uptake in the kidney. And with this compound, actually, we are now in preparation of going into clinical trials. I would like to switch gears and show something else which we are doing in our laboratory. So mainly at the Paul Scherrer Institute, we're looking for new radioisotopes which could be used for theragnostic application. And terbium is one of the few elements of the periodic table where we have different isotopes which can be used for all indications used in nuclear medicine. We have an alpha particle emitter, this terbium-149. We have a PET isotope, the terbium-152, and you have a SPECT isotope, the terbium-155, as well as a beta minus and Auger electron emitter like terbium-161. And we have, locally, we have already been introduced by Auger electrons. I call this terbium label like the Swiss Army knife, because you can really do with one element all the indications. And I think it's important to know, because if you switch from one isotope to another isotope, if you do, you basically have to restart all your kinetic modeling, your pharmacokinetics. But if you stick to one of the same element, you can actually do predictive imaging with before you do therapy with one of the same element. So lutetium-177, that was mentioned a couple of times. This is currently the gold standard in the clinics for therapy of neuroendocrine tumors, but also prostate cancer. And terbium actually features quite a lot of similarities in terms of half-life, beta minus energy, photon emission. It also oxidation state, the preferred chelating system, and also the routes of production are quite similar. However, the difference is really in this conversion in Auger electrons, and it has been mentioned before. So a beta minus particle has a relatively long path till it it can travel several cell diameters, so it can be used for medium size to small size metastasis. Whereas Auger electrons, which are emitted from the electron shell, from the nucleus or from the atom, actually have a very, very short path length. So you can almost use them for single cell killing. And if you bring them very close to DNA, at least that's textbook, they also can lead to a double strand break. So we could actually show indeed that this terbium versus lutetium has a higher therapeutic efficacy. So these are an antibody which is labeled with this terbium versus lutetium. You can see the tumor growth is significantly delayed with terbium-161 compared to lutetium. And also with a folate acid derivative, we could show that the overall survival of tumor mice was significantly enhanced when you use the terbium-161. We also performed the first in-man study in collaboration with a German group of Professor Baum, and he labeled somatostatin analog, so DOTA-TOC, targeting the neuroendocrine tumor or somatostatin receptor, and he could show with this isotope again the lesions in this liver metastasis in this patient. And basically, this was a subtherapeutic dose of the isotope. And nevertheless, it shows that with subsequent lutetium therapy of the patient, that this biodistribution matched completely that of the Terbium-161. So with that, I hope I could convince you that radiopharmaceutical development is really a multidisciplinary endeavor. So we need physicists to develop the target tree. We need radiochemists to separate the new isotopes, so develop new isotopes, the biology, the pharmacists to develop the compounds, and the nuclear physicians to apply it in humans. I think radiopharmaceuticals are indeed prototypes of theragnostics because really can structurally unaltered compounds can be used for diagnosis and therapy. It is still a domain which is preclinical as well as transitional research can be done in academic setting, which I think is very advantageous. New targets are currently investigated. So it's, for example, the folate receptor in our group, but also fibroblast activated protein targeting molecules are under its investigation, or minigastrin receptor targeting molecules, which we're also performing studies here at the Paul Scherrer Institute. And last but not least, I think new radionuclides with new features, new decay properties have made the distinct differences in the application. And I think it's very important that we look into the right combination of isotope and targeting molecule. With that, I would like to thank you for your attention. I'd like to thank all the people which have been contributing to do the work, also the funding agencies, and also the industrial collaborators we have. And last but not least, I would like to thank my entire group for their support, and I'm happy to take questions in the Q&A session. Thanks very much.
Thanks a lot, Roger. Thank you. That was a great talk. Yes, Christine?
Perfect. Yes, thank you, Roger. Fantastic to see clinical translation again, really impressive. And thank you for working on ovarian cancer, which is often a disease that is overlooked. I look forward to the Q&A session. So our next speaker, very exciting, Professor Gang Zhang, who is also a senior scientist at the Princess Margaret Cancer Center. His group is developing next generation tools for better imaging and developing more targeted, less toxic, and more effective cancer therapies. Using cutting edge technology and a highly collaborative approach, they are exploring the application of porphosomes, which are non-toxic nanoparticles that can safely travel through our bloodstream, accumulate inside tumor cells, and degrade in the body. And the title of his talk today is Porphosome Nanotheranostics, from Discovery Towards the First in Human Trials. Welcome, Gang.
Thanks, Christine. Can you hear me all right?
Absolutely perfect.
Okay, great. So super exciting to see my two colleagues, Roger and Ray, was successful in the clinical translation. And we are still fighting. And I'd just like to share our lesson on the nanotheranostics, good and bad. A lot of a painful lesson, but I think it's very valuable. So my talk is about porphosome. So what are the porphosomes? So about 10 years ago, we discovered a phospholipid. When you have a porphyrin ring replacing one of the fatty acid chain, it should become a porphyrin lipid. And this porphyrin lipid can self-assemble into a liposomal-like structure. It's called porphosome because they're similar. And the fact is, when you have 100,000, 80,000 of porphyrin, each light-absorbing molecule packed tightly into a bilayer structure, it actually absorbs tons of light and without any emission by the fluorescence because everything's quenched. The consequence of that, it is a very efficient thermal conversion agency. So we take advantage of that to develop photothermal therapy and photoacoustic imaging. And the nature of the porphyrin as a metal chelator, because it's just like the magnesium in the chlorophyll, or iron in the hemoglobin, in the heme, in the blood, the porphyrin is a very nice metal chelator. So we actually use the copper 64 for the PET imaging label and replacing using that manganese, a paramagnet metal, you combine with the massive amount of the porphyrin, you give a very nice MI contrast. When the porphyrin dissociate, it returns to the monomeric form. That is when you have a photodynamic therapy and the fluorescence features. Porphyrin like liposome, so you can do the drug loading just like the liposome does. You can also target it. So over the years, we have demonstrated the porphyrin's multifunction utility in many, many different tumor models, tumor species, and the animal species and cancer type. We actually did a model from all the way from mouse, rat, hamster, rabbit, dog. And this is just one example to show the multifunctionality in one single model. That is the VX2 Bruker carcinoma rabbit model in oral cancers. If you look at this pre-operatively, when you do the PET imaging of the copper 64 porphyrin, you can not only see the tumor lighting up like a fireball, but also see the drainage, beautiful drainage to the, essentially to the lymph node. And these are the metastatic lymph node. And this pre-operative imaging by the PET really excites us. And then we want to see how that works in the pre-operative, in the intraoperative setting by the fluorescence guidance. Indeed, we actually observe the same effect. You can see the tumor shining behind the skin, which is a little fuzzy. But when the skin was removed, the tumor bed was reviewed. And these during surgery, surgical bed got completely excised the tumor. Then going deeper, you will see the lymph node, which is the metastatic lymph node. And what's interesting, if you look further, this migration of the lymph node to the second lymph node can be observed. So the surgeon is very excited about this because not only you can do the preoperative planning where the tumor and the lymph node drainage is, you can also in the intraop setting to do the fluorescence guide resection and all the way to find the last lymph node, which is the fluorescence showing the tumors. The same principle, we actually do the MRI. You can see the drainage can be removed, basically reviewed. This drainage can be reviewed by under the MRI scans. And what's most exciting about this porphyrin cytotoxic nature is you can do the phototherapy with it. In this case, the same agent without injecting a second one, you can look at the 2 centimeter tumor in the tumor can be completely ablated by the photothermal ablation, lead to the tumor cure. So this is just a demonstrated example of the intrinsic multifunctionality of the porphyrins. So over the years, we have now keep adding different functions into the porphyrin. Now it becomes a major family of the nanomedicine. For example, by changing the way how the porphyrin lipid building block self-assembled, we can make the particle in different size, shape, and different function. For example, the HDL-like, high-density lipoprotein-like mini porphyrin, which is mimicking a lot of feature of HDL, has some many interesting applications beyond cancers, especially in the cardiac. We actually developed a stimulate responsive J aggregation porphyrin, the porphyrin nanoemulsion, which incorporates the CT component. We have developed a major effort on the porphyrin microbubble for ultrasound application, particularly in its potential for micro-nano conversion for the drug delivery. The giant porphyrin vesicle as artificial cells, developing. And the next second approach is everything we do is about this porphyrin. But if we're replacing the porphyrin with another dye, can we still form porphyrin-like? And the answer is yes. We're able to form a bodepisome by replacing the porphyrin with a bodepi, which is a very nice fluorescent dye. We can also using this one for the RNA delivery. But the most interesting part related to this session is the concept of making the porphyrin replacing with expanded porphyrin is called nanotaxophrin. So in this context, taxophrin is a porphyrin, but we expand ring. The feature of that is instead of porphyrin has a 4 coordination site, the taxophrin has a 5 coordination site. Not only is that, you have 20% larger metal chelating central pocket. When this taxophrin lipid self-assembled into a porphyrin-like structure, when we call nanotaxophrin, it actually generating a lot of interesting possibility because now so many different metal, particularly lanthanide metal, especially this Roger and Ray mentioned, all these beautiful medical radioisotope can be starting to incorporate, like yttrium, indium, bismuth for the alpha, lutetium, thallium, and terbium. Now I learned the terbium from Roger. We will look at this one as well. And thallium, we actually now work with Ray and Phil Bauman in the KCL, are doing the AOG electron, introducing into the prophesons. So just for example, test, we actually show we can titrate in the radioisotope indium-111 with a cold lutetium into this nanotaxaflame, and together with a PSMA targeting ligand, collaborate with the Marty Pomper in Hopkins. And we radio label them through our own microfluidic chamber platform, and achieving very nice radio labeling yield and radio labeling purity. And once we apply that in animal model in a PSMA positive negative tumor, we can actually see both by the fluorescence imaging of lutetium-175, which is cold, and indium-111, which is hot by SPECT, you can observe the consistent tumor targeting. And that lead to very nice photodynamic therapy cure efficacy by this cold lutetium. So we believe this open up new floodgate of bring the prophesone into a radiocyanotic space, and we will so much radionucleotide we can take advantage of. So my talk focus actually today is really on the lesson we learned, both good and bad, on how we actually move prophesone to first in human. And since its discovery 10 years ago, it's already 10 years past. So I want just to give a few points. One is identifying the suitable clinical scenario that can take advantage of prophesone intrinsic multifunctionality. That is a very key in terms of developing, especially nanomedicine into clinic. You have to have these scenario, which is the most translatable. Second is a challenge of radioisomers, and we learned a lot on that. And the species factors, we learned a lot. And finally, the good part, the prophesone-enabled hybrid phase 1A and 1B clinical trial design, which we like to share with you. So first, the minimally invasive, the transbronchial imaging guided photothermoablation is we have in mind for the lung cancer application as our first clinical scenario. Because in early stage, the current standard is anatomy, essentially just a resection by the minimum, so-called minimally invasive surgical resection in the lung. But many patients are ineligible. Like for example, these patients had the chronic lung failure, like the heavy smokers. They're very difficult to do surgery. So even their early stage, they actually have very major risk because they can't even do a surgery with it. So that become a very unmet clinical need. So our idea is we need an optimal treatment strategy can have very high efficacy, which lead to improved cancer control and high specificity, which is minimum off-targeting toxicity effect. So to work with that, we actually developed a rabbit model with orthotopic lung cancer, because rabbit is about like 4 kilograms, like a baby. And we do have access to the Olympus pediatric bronchoscope, which can fit in for this purpose. Once we have the tumor inoculated in the lung, as is showing here by the CT, we actually see the tumor is actually encroaching from the bronchial side. So essentially the tumor is behind the bronchial wall. Once the model is established, this is the method we're going to take. We want to do minimally invasive way. So we designed an approach to do the transbronchial. So instead of go transcutaneously, like most of therapy do, we want through the bronchoscope, take advantage of the modern advancement in the bronchoscope, like robotic bronchoscope. So we want to see, bring the scope next to the tumor. Once the prophesone lighting up the tumor, through the bronchoscope, you should be able to observe them. Once the tumor site is delineated to identify, then we can insert a fiber. We can do the ablation right away. And because it is bronchial, they're actually much safer because you don't risking of deflating the lung. So this is our idea. So to show this is actually approach actually working at proof of concept level, we demonstrate here, and this is a deep in the bronchial wall. and we're using our own bronchoscope modified for photothermal and fluorescence, we can observe there is no, but 45 minutes later after post-injection, there is no fluorescence shows up. But 40 hours later, when the tumor is imposed, you can see there is a two tumor, 1 and 2. They are behind the wall, right? So this is actually, you have to look carefully to see the tumor behind the bronchial wall. The reason why it is moving is because rabbit is breathing 2 times faster than human. And this really underscores if you don't see the tumor, because now I can see the tumor. Now we can do the ablation, and we observe a very good selectivity between the pulfosome ablating the tumor lung versus the normal lung tissues. So the next step is, because the VX2 is a rabbit tumor model, so we want to see the biological relevance of this approach, because we have to use a human cancer cell line. So for this, we developed, together with the talented Japanese surgeon, we developed this oral garage method through the vocal cord instead of doing the surgery, seeding the tumor directly to the lung and generating orthotopic tumor in all 3 major types, A549, H460, H520. All of them show the nice tumor selectivity of the pulfosome from the non-tumor to the normal lung. So the biological feasibility is validated. The next step is to technical feasibility, because we want to bring the technique to the human. The closest to the human lung is a pig lung. So, but pig doesn't grow tumor easily. So we actually, in order to make it more relevant, we're using the mouse tumor to pulfosome injected IV, slowly accumulate tumor. Once the pulfosome in the tumor, we take the tumor out, then seed it in the different location of the pig lung, which allow us to do the transbronchial fluorescence imaging, transbronchial fluorescence guided biopsy, and transbronchial photothermal ablation. And this basically validates the technical feasibility of this approach using the pulfosome-enabled transbronchial imaging guided ablation. But this is not enough, because for clinical to be used in the truly in the clinical translation setting, current bronchoscope only go to about 2/3 maximum into the lung. So that means there is a lot of these tumors you cannot be reached if we're using the bronchoscope. So to make them truly useful, we can team up with the Japanese fiber companies. We actually developed these novel fiber scope, the laser scope, 1 millimeter, less than 1 millimeter diameter. You can have 2 channels, one for imaging, one for therapy. Equip this such ultra thin fibers, because the bronchoscope cannot reach is where the fiber can reach. We can reach 6 centimeters deeper, 6 centimeters deeper. This allows us to basically reaching the entire lung. So now we actually solving the technical feasibility, especially if you combine with robotic bronchoscope. In theory, the technical hurdle to apply pulfosome for lung cancer transbronchial imaging guided photothermal ablation is ready to go forward to the clinical trials. So speak of clinical trials, very challenging process. I want to share 2 lessons. One is the radioisomers. Many years ago, when we discovered the pulfosome, we actually find out pulfosome is assembled from porphyrin lipid. And the porphyrin lipid has 2 isomers, SN1, SN2. And these 2 isomers, we can actually nicely isolate them by the enzyme enrichment process. We even have a patented process for that, so we can have both pure isomers. What we find out, these isomers doesn't impact the pulfosome's function, structure, function, everything maintained, except the SN2 isomer has a very nice biodegradation. By 24 hours, we can see in the liver, in the spleen and the liver, they pretty much all dissociate. But in the blood, they remain intact. That means the pulfosome in the blood is stable. They can reach in the tumor site, going their own accumulation process. But the liver spleen, they can actually completely degrade it. So this way, we can actually get a problem. The Health Canada tell us, well, you need either fixed isomers or a single isomer, and they need to be scaled up. That takes us 3 years to accomplish. We got this 200 milligram scale now. 95% purity of SN2, and the yield is 85%. We confirmed the isomer in terms of metabolic studies. We did a toxicity study in the mouse, rat, dog, and the monkey. I think I used all of my time, so I will not explain. The major issue we actually learned is the dog is very sensitive to PEG lipid, so as a pig, but the monkey is not. If we actually we learn the lesson. Next time if we do it, we will not using the pegylation, we will not use the dog model. We will go straight to monkey. That will solve all the problem because monkey is very close to human. Dog has a PEG sensitivity issues. So finally, porphyrome's intrinsic multifunctionality does allow us to have one unique advantage. We can design a phase 1 trial under copper 64 with a low dosing to be more safety. Then we can instantly, once they're safe to infusion, we can apply that to all 4 different cancer sites using different complementary function by the sheer clinical trial infrastructures. So essentially, this is a lesson we learned. We planned clinical first in human in 2009. We're now 2 years after. We're still not in patient yet, but we are really getting close. All these steps has been accomplished. And hopefully one day, very soon, we will be like our colleagues, my previous speakers, will get into the patient, and not just for imaging, but for the true therapeutics. So finally, I'd like to thank my group. Particularly, I want to single out Michael Valek, who has been instrumental in driving the translation for the last 7 years. So a big thanks to him and to all my students, my collaborators, my fundings. Thank you.
Wonderful. Thank you, gang. Thank you for giving us some insights into your journey to clinical translation of the porphyromes and some of the lessons that you've learned. It's really helpful to all of us. So I think we will open this now for our Q&A session. So please feel free to either raise your hand and Christine Mesquita can allow you to speak, and or just add your questions into the Q&A panel. Maybe we can get started, JC. I wanted to ask a little bit about, we see this is all very, very impressive research and important research. The reliance on things like MR-guided focused ultrasound, PET, SPECT imaging, thinking about availability of radionuclides. Can you speak to us a little bit about how accessible this infrastructure is, these radionuclides are, and the extent to which it may be realistic in the future for there to be widespread adoption of these types of approaches?
Do you ask me? Sorry.
Sure. Roger, Ray, gang, anyone, just trying to understand, we're both living in major sites, right? We have the good fortune of being next to cutting edge clinical sites. So I think we're very lucky that way. But to what extent has access to these types of, this type of imaging equipment, clinician scientists with this expertise, to what extent has that improved maybe in the last decade, for example?
Well, maybe I can speak for the European situation. I think at least in Europe, these sites for performing imaging is quite good. I mean, there's most of the hospitals somehow have a nuclear medical department. They have capacity to do imaging with PET or SPECT. And therapeutic applications, I guess also there you need a certain specific infrastructure, mainly for managing of the patients, and you find those likely in larger hospitals. So I think it's not really such an issue. I mean, what we are very fortunate in Zurich is that we have, and also the Paul Scherrer Institute, we have access to facility where we can do GMP production of radiopharmaceuticals without having the pressure to do the routine clinical work besides. Very often this infrastructure, because it's very expensive, actually it's integrated into a department of nuclear medicine. And of course you have to take care of the patients which come in on a routine basis. And then for research, there's very little space or time and also money left. So that is a bit of an issue. But I think the infrastructure in Europe is not so bad. It needs a bit of trained people to bring new things to the clinic. The routine is quite well established, but maybe also Ray can say something about the situation in Canada.
Thank you.
Yeah, so in Canada, SPECT is everywhere. PET is not everywhere. PET is mainly at the major teaching hospitals in Canada, but it is a widely used technique, and SPECT has been used for decades in nuclear medicine. In terms of the MR-guided focused ultrasound, that's a very specialized technique that involves MRI imaging, and this may not be readily available everywhere. But I think, in terms of the radioisotopes, indium-111 is widely available. Copper-64, some other fluorine-18 PET isotopes are relatively widely available. Actinium, there is actually a shortage in the world right now of actinium-225, and there's a lot of interest in developing actinium-225 labeled radiopharmaceuticals. So this may pose a problem in the future to widely apply actinium-225 to targeted radiotherapy, although a number of scientists are actually developing different strategies to produce actinium-225, including a cyclotron-produced radioisotope, which may make it more readily available. So I think, at this stage, these are sort of techniques that are being explored at the clinical research level.
Mm-hmm.
How they will be adopted by the healthcare system in the future remains to be seen.
Mm-hmm.
Yeah.
That's great. It's an exciting time. Thank you. Ray, we do have one question for you. So this is a question, I don't know who this question is from, but it's the work is very nice and the presentation very interesting. Is MRI focused ultrasound painful for the patient? Do they need anesthesia?
No, they don't need anesthesia. It's not painful for the patient, and actually,
because of this.
Sorry, I'm hearing something. So it's not painful for the patient. Patients aren't under anesthesia undergoing this procedure. However, the safety of MR-guided focused ultrasound is also being evaluated in the clinical trial. It has actually been applied for a number of other clinical situations to try to improve drug uptake into the brain. But there is potentially some adverse effects from MR-guided focused ultrasound, which could occur and is being evaluated in the trial. For example, hemorrhage in the brain, et cetera. But so far, it seems to be very safe and can be combined. I also want to make a point that actually in the clinical trial that I described, the imaging is actually not intended to ultimately be a clinical tool. It's actually an experimental tool at the moment to evaluate delivery of trastuzumab to the brain in the patients, combined with MR-guided focused ultrasound. And as I mentioned, the patients are only receiving the imaging agent on the first treatment study, not on the subsequent cycles. So it's just used as an experimental, not an experimental tool, but an imaging tool to evaluate drug delivery. So I think this is another thing we also have to keep in mind, that not every application of radiopharmaceuticals is a clinical application, but in some cases it might be used as a tool.
Right, thank you. Gang, I can see you have your hand up. Please go ahead.
Yeah, just a quick question on the Ray. So the focused ultrasound together with this radiopharmaceuticals is not the first time you put a radiopharmaceutical into patient, right? Because the same agent, only when you're getting the first in human experience or the track record, they will allow you to combine with the more sophisticated MR focused ultrasound study, right?
Yeah, I mean, imaging studies with radiolabeled antibodies have been, done safely in patients for many years. What we're doing in this particular example is using the imaging with the radiolabeled trastuzumab as a tool to evaluate whether focused ultrasound will improve the delivery of trastuzumab. So But we still needed to go through the Health Canada submission to, because this is a novel agent that we developed, a radio-labeled form of trastuzumab. So we need to, assure the safety of the radiopharmaceutical, both from a pharmaceutical point of view, pharmaceutical manufacturing point of view, as well as preclinically. A lot of the studies that you're having to undertake right now for the porphysomes.
Yeah, so I just want to emphasize it is really each step. You're not like go to the home run. Each step you have to overcome before you go to the next step, right?
Yeah.
Actually, sorry, I have a quick question for Roger. Is a terbium 161 is readily available?
Well, yes and no. I mean, we produce it. We have now performed the separation, and it can be, it's performed in analogy to lutetium-177. So it's gadolinium-60, which is irradiated in a nuclear reactor, reducing gadolinium-61, and then this decays to terbium-161. Actually, yes, we're producing now gigabecquerels of this isotope. We are scaling up. The quality is very similar to the lutetium-177. And we are negotiating with other groups and also companies to make this isotope available for more widespread indications, applications actually. But for preclinical collaboration, I think with the 7-day half-life, one could even ship it to Canada.
There we
Thank
go.
you. Thank you.
I see terbium labeled porphysomes in our future. Fantastic. Roger, you had your hand up as well.
Yes,
and then we'll
actually,
go to Cornelia. Thank you.
Yes, actually, I have a question regarding the tox study to Ray. Did you have to go back to do tox study on your functionalized trastuzumab? Or could you just manage it without saying, okay, we have low quantities and we do everything on the GMP?
Yeah. So one of the strategies that's proven quite successful for translational radiopharmaceuticals in my group is to actually use an already approved pharmaceutical material to construct the radiopharmaceutical. So this agent is based on trastuzumab, which is an approved drug in Canada. And so the issue with Health Canada in demonstrating safety is always just for an agent that's based on an already approved drug is to demonstrate that when we modify it for radio labeling or whatever other modifications we do, it doesn't increase the toxicity of the drug. So we did have to do some preclinical toxicology studies, but only in mice, not in a non-rodent species.
Oh, that's great, because otherwise, I mean, this is just out of possibility for academia to do any monkey studies to do this. And actually, I have a second question regarding this gadolinium DOTA. When you open the blood-brain barrier, I mean, is there any issues with toxicity? Because this gadolinium, basically the blood-brain barrier basically closes afterwards. Is there any, gadolinium could be trapped somehow in the brain?
I'm not sure, but as I mentioned, the safety of the entire procedure, including the application of the focused ultrasound, as well as using the contrast agent to evaluate blood-brain barrier disruption, as well as the safety of the radiopharmaceutical. All of this is being evaluated in the clinical trial. And so far, actually, I showed the results of 4 patients, but we've actually done 7 patients so far. And we haven't had any serious adverse effects in those patients.
Just as a side remark, ETH is currently developing a head PET camera, very, very thin, so you basically could include a PET insert into your ultrasound system and actually would not have to use actually gadolinium, but it could use a radiopharmaceutical, to see, penetration into the opening of the blood-brain barrier.
That's very interesting, because actually we're planning to extend this imaging approach to another antibody with MR-guided focused ultrasound and develop a new radio-labeled radio tracer. And that radio tracer will be zirconium-89 labeled antibody for PET. So we only use the indium because we had already developed that agent for another purpose. So it was applied to the clinical trial, but I think PET is probably better. And also there's PET MR systems now as well, right? So MRI is, common imaging technique for the brain. So it's good to combine PET and MR.
Wonderful. Thank you, Ray. Thank you, Roger. And our last question or comment to Cornelia. Please go ahead.
Actually, I also have a question for Ray on that beautiful talk on focused ultrasound. So, I mean, I see the beauty of using an already approved antibody, but I was just wondering whether size also matters in your case, and if one could do the same job with a fragment. I don't think trastuzumab really needs an FC part. It's a big molecule. Do you know whether opening up the blood-brain barrier would benefit? I mean, whether delivering a smaller agent would be better also for the penetration than in the outside of the vessel for kind of hitting all the tumor cells? Or any experience on this? How much size matters?
Yeah, I don't have the answer to your question, but the only thing I will say is that in this particular clinical trial, what the group at Sunnybrook wanted to do was radiotrace trastuzumab. So we had developed for another clinical trial, which never actually got started, this indium 111 labeled form of trastuzumab, the intact antibody, and it's probably the closest we can get right now to tracing trastuzumab. However, as you mentioned, perhaps for further development into a radioimmunotherapeutic agent rather than trying to trace trastuzumab, a smaller fragment might penetrate better than an intact antibody in that situation. I don't know. Actually, it's a good question.
Wonderful. Thank you. Well, I think that brings us to 12 o'clock. I just want to thank our 6 brilliant speakers and our engaging participants today. What a wonderful symposium. We can certainly see lots of opportunity for collaboration and partnerships. I encourage you to reach out to one another, including our participants. You saw something in one of the talks, please reach out to our speakers. And if you need our support with that, please reach out to myself or Christine to let us know, Christine Mosqueda, to let us know how we can help you. So thank you so very much, everyone. And we look forward to seeing you soon. And a very special shout out to my co-host, Jean-Christophe Leroux. What a delightful surprise for me to be able to spend a few hours with you today, JC.
Thank you, Christine. That was a great symposium. Yeah, I really enjoyed it a lot. Thank you to all speakers.
Wonderful. Thank you, everyone. Take good care.
Thank you. Take care. Bye-bye.
Thank you.
Bye.
Bye-bye.